Good morning, everyone. I'm just going to work on getting some logistics set up. It's lovely to see you all here. Um, so feel free to come off mute and chat or stay on mute and hang out until we get started in about 10 minutes.
Oh my, I look rough.
Good
Hi,
morning.
morning. Most of it until you need me, I'll be on mute and um my camera off.
That is absolutely fine. You could do that the whole time if you would like.
I don't know, but because my father-in-law's not here yet, so
That green in the background is,
I'm
um,
a little princess over here.
oh,
Right now she's watching Wicked.
sure. A favorite in our household as well. Good morning, Carolyn. You are still mute.
No, you're
See if
on mute
you're on mute.
on the left
There.
bottom.
There you are.
Oh, sorry. I um I am Lauren O'Donnell's mama, and I just want you to know how much I appreciate you and your strength. And I can see that you're in the hospital now. I have
So
my I'm in chemo, so I got my fancy chemo hat on. Um, but Lauren and Nick are also in the hospital with Sweet Emery, and you probably know that. And I don't know, I just have to shout out some energy to you people who it's just, I don't have any words. That's all I have.
well, thank you.
Yeah,
Yeah.
I love the hat. We are very lucky here to have such a great team. And we work hard to be resilient. And so we just, roll with whatever we need to roll with and jump in wherever we need to jump in. And um, that our foundation runs that way, I think is a reflection of the strength of our community.
Yeah, it's amazing. It's an amazing community. And I know, how much Lauren and Nick have rised or, to the occasion so many times, and these warriors, they are amazing. And it's just, it's just mind blowing.
We love Lauren and Nick and Emery, of course.
Oh yeah.
I'm so glad that I get to like have more meetings with them and get to know them better and get to see Emery
Yeah.
more often. And that's really the coolest part about
Yeah.
doing what we do here.
So yeah, I was anyhow, so I'm probably gonna these hats get hot, so I'm probably gonna cover my camera and but I will be here and you guys just know that if you need things, I can do, uh
We love how active you are on our social media. You're such a cheerleader.
Yeah, we've mentioned that before. Yeah, we love it.
okay, well, I'll keep it up. It's been a little rough with some rough chemo things, but my last one is coming up here in November, and hopefully that'll take care of it, and I'll be on the mend. So, but anyhow, Nikki, I just
You're
worship you
too.
and your husband.
You.
So look forward to the meeting.
Yes, thank you.
Welcome to everybody. We'll get started in a few minutes. In the meantime, if you want to double check that um, your name is how you would like it to appear, on here, that'd be lovely. We have it set so that everybody sort of enters in on mute, and that just helps us make sure that if people come in and out, we're not disrupting the presentations a little bit later today. Um, so we'll get started, like I said, in just a couple of minutes. We're so glad you're all here.
So do I need to mute myself or did you already do that?
I can mute you, I believe.
Or Johnny, or if you can't, I'll figure it out.
I can totally do it. I already did it.
Yeah, yeah.
I'm good. You're all good. And then if I see that you want to come off mute again, I can actually uh send a little pop-up to make that easier for you.
Done, 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 Brian.
All right. Um, I have activated our translation service, and you might have gotten a little pop-up that asks you if you want to um, allow that. And if you click yes, then you can get um, automatic closed captioning through the Wordly app. In a moment, I'll also share my screen and put up a QR code that you can scan if you would like to have audio translation. So if you click within Zoom and click yes, then it's closed captioning and it'll appear on the side of your screen. If you would like to um, have audio, you'll scan the QR code that I'll put up in just a moment. And through your phone, you'll be able to select for audio translation. And Nikki, if you happen to have the QR code, um,
Yeah, send it to her.
you already took care of that. Great, thank you. Uh, mm, all right. Let me go ahead and share my screen. And we can get started with some logistics and things like that. Welcome, everyone. We're so glad that you are here. It's so lovely to see so many familiar faces. Um, if you want to start off by um, naming yourself or renaming yourself, you can feel free to put your warrior's name in there or anything else. You would do that by selecting the three little dots in the box that shows your picture. Um, just some meeting logistics. We have the meeting set up so that you'll sort of enter the Zoom on mute. We're not quite big enough to have like a webinar type situation here. So we decided to keep it a little bit less formal. And so that's why we're kind of all in the same place at the same time. So please try to stay muted unless you're speaking and asking a question. I can always mute people or ask you to come off mute if you're having trouble with that function. For translation, in just a moment, I will go ahead and um, send another link through Zoom so that you can get the closed captioning in there. You're also welcome to either use the link here or this QR code, which will come up on your phone and allow for both closed captioning, so written out translation into whatever language is best for you. And if you use the QR code, it will also allow you to um, have spoken translation, so you can sort of listen in on your phone. Um, please put messages into the chat if you are struggling with that, and we will um, help you out. As I mentioned, please add your preferred name to your Zoom box and feel free to use the chat to interact with one another, say hello. I know there's so many familiar faces here today. It's so lovely to see you all. This little QR code will occur on the next few slides as we get going here today, and that'll just help us sort of stay all together and have access to um, that translation should it be necessary. So here is a little bit of an agenda for today. Um, so we have our welcome right now with me, and we'll do a couple of activities to sort of prime our day and start ourselves off. And then we'll get to why I'm sure you're all here. These science updates from our researchers, including Dr. Albino, Dr. Ortiz Gonzalez, and I have a recorded update from the Boj Laboratory because they were unable to attend today. We'll then get into our research opportunities. So, the different things that we as a community can get involved with to help our researchers out and do um, sort of be involved, help our community, help understand TBCK syndrome better, and sort of move our way forward. After that, we will have some reports, just what the TBCK Foundation has been up to this year, what we're looking forward to in the year ahead. And we'll also have a special report from TBCK Puerto Rico, since they're so active and such great partners and provide so much to this community. We'll then sort of wind down our time together with the launch of our new Ready for Research campaign. I hope that you've seen throughout our uh, email campaigns that we are launching this new campaign. We're ready for research. I'm really excited to have Lauren share that with us, and then we'll wrap up around 2. I'm going to end the show and stop my share for just a second as I double check the chat and share the Wordly translation again with all of y'all. So you might have seen a little pop up for those who have joined us more recently. Um, and that'll just allow you to experience the translation service should you need it. I'm assuming that someone would have stopped me before if you couldn't see my screen or any of the sharing that I just did. I'm glad to see Madison giggle about that just a little bit to make sure that I was all good there. I realized that about halfway into those introductory slides. Um, Now, as people are still joining on, I'm just gonna pause for a minute to honestly check my phone and ask if Nikki would be able to post the Zoom link and the passcode onto our social media. People are asking for that.
Okay, yeah, I've been doing through Messenger. Do you want me to do it through our community page or our?
You could probably do either. I'd be pretty confident with either at this time.
Yeah.
All right. Now we're gonna try a new activity. I am going to share my screen and we're going to use something called Mentimeter. And this is a way for us to all interact together and sort of see who's here and think about what we want to get out of today and you know what we're all doing here. So I'm going to share my screen again and hope that this works. Sorry. It works out better if I start on the first slide. I know some of you have asked, we are recording this meeting so that our friends who couldn't be with us to begin with can check this out later. Um, if you would like, you can go to use your phone and go to menti.com and use this code and join this poll. We are interested in what your hope is for today. So give us a couple of words on why you're here. Um, you can also scan the QR code and it'll take you there. At least I hope so. And I'm hoping that, yes, wonderful. As more words are added, we'll keep going with this um, word cloud. And so this shows us sort of what our interests are. If you're looking at this screen and you're like, man, I'm also here for engagement and a research update, go ahead and scan the QR code, add those words. It'll make engagement bigger. And so we'll see, you know, really why we're all here. Don't be shy to use the same terms, the same language, or put your own language in here. Um, this looks super great. I'm glad that we're here for research updates and to learn together as a community. We're happy to share our knowledge about TBCK syndrome. We're happy to build connection. Our community is connected and strong, and that is so important to what we do and how we move forward as a community. I love seeing community engagement up here a whole heap and knowing how much research and updates on our research are coming. I'm excited that we're gonna um, meet that need and meet that hope for today. Um, and I love how this keeps changing and updating, and the big words we're seeing are connection, um, feel free to come off mute and give any thoughts that you might have about this. We'll give it just another minute or so. Yes. Can everyone else see? Like we're sharing the screen, we're seeing all of these words that are representing what we're feeling today, what we're hoping for today come up. Yes?
Yes.
Thank you. All right. This looks amazing. Um, I sent this to Nikki yesterday to ask what she thought about these questions, and her response was, "We're gonna have to save this so that we could talk about it later." So our results from here will be saved. Feel free to add more if you would like, but I am gonna move on to our next slide, which is a little bit more focused on our foundation. Since we have you all here together, we're going to take this opportunity to sort of find out what we're doing that y'all really like, find out if there are other things that we should be doing or could be doing that people might be interested in. I'm hoping that I did the settings to allow for you to vote for more than one, to hopefully make this not such a tough decision. Because me personally, I don't know that I could like pick between some of these. Um, I love the Meet Our Warriors. It's so great to see how our community is sort of um, getting bigger and oh, it says you can only vote for one. Oh no. Well, I'm sorry that I made it so challenging. Thanks for letting me know, Cindy. I'll have to um, try harder next time when I do this. Um, but this just gives us a good idea of, what we're doing that the community really likes. I agree that the shirts that Nikki designs are amazing and I love them. My biggest problem is that now there's so many amazing designs that I have to like pick and choose which one I'm going to wear. I went old school today with my Ask a TBCK Scientist shirt, which is one of the very first shirts that Nikki made for me. Um, but I'm also excited that she'll be putting out some new designs here in the coming months. We'll even have something new for Rare Disease Month. So we are gearing up for that. I also love the sticker campaign. It is my family particularly loves sponsoring that activity. If you have ideas for sticker designs or things like that, go ahead and send them my way. We do a little bit of a mix of what we're able to get on discount as well as new ideas that come through. And if you're interested in the PR parade, go ahead and send a message to Inez at tbckfoundation.org. She and Gina are working on that. They've already started planning for this year. And so if you're interested in getting involved with that, go ahead and reach out. And I'm sure they'd love to have more people. Our Meet Our Warriors, Amber heads that initiative for us, and she's letting me know which ones I haven't added to the website yet. So if you have done a Meet Our Warrior and you're not on the website, fear not. Amber is keeping me in line and making sure that I get those up quite soon. Um, and our mental health Monday initiative is also a team up between Inez and Amber, reminding us all to take care of ourselves as caregivers and take care of one another as our community. All right. Thank you to everyone who has responded. Um, and thanks for, I guess, listening to me chat for a while. Um, so this one will pop up some words for us to think about. I added this question in here because the other afternoon, my 5-year-old came home from school and said that she wanted to play with her dad and that she was going to be a TBCK doctor because she was going to help people. So, if to her TBCK means helping people and being a doctor, I thought that I would love to hear what other people come to their minds when they hear TBCK. I guess this also lets us know if we're repeating things enough. Uh, our administrative director, Karen, is always telling us that we need to repeat the same things over and over, that repetition is the essence of education. So we hope that you don't get bored seeing the same things from us over and over, but we know that it's important for growing our community. Just gonna check the chat real quick. See if I've missed anything. Oh, Michael added family as something that he thinks of when he hears the word TBCK, which is also a great one. If anyone else is having trouble with the Mentimeter, feel free to add it to the Zoom chat here. Um, I love the idea of TBCK uniting hearts, homes, and labs. Oh, Dr. Ortiz Gonzalez added mission with an exclamation point in as something that she thinks of when she hears TBCK. And I feel that that is a reflection of her passion and dedication to TBCK research in the lab. All right, so this sort of aggregates our ideas into a couple of different modalities, which is pretty cool. I am going to check the chat again real quick. We look pretty good. All right. Thank you all for participating in this activity, showing us what we think about when we think TBCK, helping us see uh, where we're doing things that are appreciated and um, what things the foundation can be doing more of, and helping us sort of align and get ready for what's going to be a really great day, I think. Um, if you have any questions, feel free to unmute yourself and ask. I'm just gonna stop my share real quick and switch to our meeting logistics just so that I get this Wordly QR code back up just in case anyone who has joined recently and needs translation. Let me know if that is not working for y'all, and we will go ahead and figure it out. The last thing I have in this sort of introductory area is right here with our infographic initiatives. So I am in the process of getting a new printing of these infographics. We've created a new QR code here. You can see over on this side, it says TBCK Gene Review. And for our friends from Puerto Rico, they're super familiar with this because a couple weeks ago they did a meeting all about that gene review. But this is the article that Dr. Ortiz Gonzalez wrote earlier this year that really just gives you all of the information you need to know about TBCK syndrome. It's a great one to share with clinicians. Um, and whenever or wherever you see them, it's a good primer on what is TBCK syndrome, what to expect. There's some guidelines in there for care of TBCK warriors, and it is a good sort of one-stop shop for all of the information that you might need about TBCK syndrome. As I mentioned, I will be printing both this larger sort of half-page infographic about our warriors. I will be updating these sections as our uh, oldest warrior recently celebrated a birthday, and our community has actually expanded to, I think, Nikki, correct me if I'm wrong, it's like 130 now.
Yeah, probably 131 now. And I was about to say too, is because we're in the hospital and at Cincinnati Children's, like there's only 35 in the world. And I'm like, no, our numbers have changed. And like, so that's why like this is important and to be here too, is to get your information, and you'll hear about that more too. Just so we can get all the numbers out there to better help our community and to better help the medical teams that serve you as well.
So this is, go ahead. This is an initiative that we have here at the foundation. So we make up these cards. There's the larger ones that are at sort of a half page. They're made of cardstock. And then it was Nikki's idea to create this business card design. And so on the back of these cards are QR codes that lead to more information, either to our website or to that gene review article. And you envision this as a tool for our community to both raise awareness, but also um, sort of take the burden of education off of our community members and sort of give you the tools you need to make sure that your clinical teams are up to date on things. As Nikki mentioned, oftentimes our clinicians have read a review a few years ago and it's not really up to date on what's going on. And so we really work hard to make sure that these infographics, the information on our website, the information in the gene review are all sort of our cutting edge, up to date, all of the information that we have. And so if you're interested in having some infographics and having some business cards, just go ahead and send me an email, emily@tbckfoundation.org, and I will be happy to pop them in the mail to you as soon as Canva mails them to me. So if you want them, go ahead, send me an email, and I will get them in the mail. And with that, we are ready to switch our gears towards the meat of what everyone's here for today. We have our science updates. Hopefully, Dr. Ortiz Gonzalez is just about ready to sort of take over and share some updates from her lab, as well as just a little bit of priming information about TBCK syndrome to make sure that we are all on the same page as we start off today.
Good morning, everyone. Buenos dias. I am sending a sharing request. Okay, now it's letting me do it. Screen share. Okay, give me 1 second. Okay. It looks good on my end. Do you guys thumbs up? Looks okay? Okay. You can hear me okay?
Yes.
Perfect. Well, thank you for having me. It's always great to see everybody and like see all the beautiful community from our TBCK Warriors. I wanted to start the day with kind of a quick overview in case there's families who are facing a recent diagnosis. People always ask me to kind of give like the big picture view of TBCK. So we'll just do that for a couple of minutes just in case we have new members to the community that would benefit from that. And then I also wanted to do Emily and the foundation asked, how are we moving forward in the big picture, and why are natural history and biomarker studies important, and what does that mean? Like kind of the definitions of those, and then I'll wrap up with some updates from the lab. So this is basically the outline of what we're talking about. I can't see the chat, so if you have questions, let me know, or Emily, you can tell me. All right, so let's get started. So always starting with the basics. People always ask me, what is TBCK? What does it even stand for? So that's what it stands for. It's the name of a gene that encodes a protein, and the protein, it's called TBC1 domain kinase. The reason why we're here is because our warriors have genetic variants that affect the function of this protein, and we're trying to figure out what is the best way to move forward, right, in terms of the disease that this causes, and how do we find better treatments for it. There's several names for the disorders, and people use TBCK. I think TBCK syndrome is sort of the easier one that most people use, but there's TBCK encephaloneuropathy, which is a term we use for the kids that have a motor neuron involvement, so a lot of weakness and neuromuscular involvement and the muscle weakness. And then that we have a very active component from TBCK Puerto Rico, and that many kids in our community share that genetic ancestry, and that is due to a founder effect. So when you hear that, just a reminder that that is basically people that share some genetic traits because they come either from a restricted geographic space, for example, like an island, or other circumstances where there's less variability in the genetic pool. But that's what the Boricua variant, you may hear the founder effect. So that's like your 101 words to make sure we're on the same page. I think these I would call the core symptoms of TBCK, the symptoms that I think most of the kids have, regardless of ethnicity, regardless of what variant. Most kids have low tone. They have developmental delay, which is typically global, meaning motor, language, fine motor, social. The MRI can have changes, and those can be variable over time. We'll talk a minute about that. And I think you know better than anyone that one of the biggest struggles for families is management of epilepsy and neuromuscular weakness. So I think there's other things that come and go, but I would call those sort of our core symptoms that most children share. Families always ask me to talk about TBCK and seizures because that's part of kind of the daily challenges again. So I wanted to kind of just put a quick reminder that the time course of the epilepsy, usually for most kids, they start having seizures in the setting of illness. So you have a flu, you have fever, it could be even fever associated with a vaccine, but the fever or the illness sort of is triggering first episode, and many times people get diagnosed first with what we would typically call febrile seizures, and this is usually less than age 2. And then we have unprovoked seizures, which means seizures without any clear triggers, and that's the definition of epilepsy, when you have multiple seizures but you're not sick, and they sort of randomly happen. I would say in my experience, over 90% of patients with TBCK variants have a diagnosis of epilepsy, especially after that age 2 or 3. And then we know that one of the biggest challenges for our warriors is as they get older, that seizures can become very difficult to manage, and you need multiple medications, and they become very, the seizures don't respond to medication as well as they did when the kids were younger. I think especially in our community, I always remind people that we treat seizures especially status epilepticus, when the seizure doesn't stop by itself. If you have a seizure that is lasting over 5 minutes, that's when we start thinking about that. We are aggressive with rescue medication because kids with TBCK tend to have a lot of respiratory problems, or like they stop breathing, they pause breathing, they end up having to go to the hospital, be intubated, need respiratory support secondary to the seizure. So that brings me to my slide is stuck. There we go. Frequently asked questions in clinic about management. We talked about the triggers like infection. Also, I always remind people low sodium can be a trigger. So if your kid's hyponatremia, it's something to keep an eye on. But also it's, to me, very important to talk to your neurologist about your seizure action plan and make sure you have a clear plan. So when you have a seizure that is prolonged, we try to avoid the best we can, having that respiratory problem of having to need respiratory support or having to be intubated. So specifically the timing of the medication, we often in neurology say give a rescue at 5 minutes. For kids with TBCK, I usually have a conversation depending on the pattern of the seizure, and if parents always know, if they know that if it goes over X amount, let's say 3 minutes, every time the seizure goes over this amount, we end up with trouble, then you could discuss, okay, why don't we give the seizure rescue at 3 minutes, let's say, instead of 5? So that's a conversation that's very kind of individualized depending on your history, right? And what is the pattern, but it's something that I remind people because it could make a big difference in terms of avoiding hospitalizations. I listed some of the common seizure medications that we try kind of in order, based on experience. This is one of the things that, for example, collecting larger amount of data, we could have a better sense, and just put a last plugin for non-pharmacological, so non-medical, non-medicine therapies, which there's been some experience with patients that have had, for example, a VNS for rescue. Ketogenic diet, I think some kids tolerate it, some don't. So I'm a little bit on the fence of how to recommend it. And I haven't heard of anybody with DVS, but if there's anyone out there, I would love to hear their experience. Next, in the sort of core features is the weakness, which you guys know it's another of the big struggles for our TBCK warriors. So I think that also tends to be kind of variable in terms of different kids with different genetic variants. And it's also variable on how quickly or not it can change over time. So kind of the progression part of it. For most of our patients, we do see that there's an evidence of weakness because of the motor neuron connecting to the muscle. So that's called a motor neuronopathy. So if you think about brain to spinal cord to muscle, that's a circuit that allows us to move. That connection from the spinal cord to the muscle, it's often affected, and we see that by when we check our reflexes, we see that kids with TBCK have very often low reflexes or hard to get reflexes. We can do fancier ways to measure that, like EMG, which is electromyography and nerve conduction studies, and we've done that in the past, and show that there is evidence again that that part of the circuit is affected. How does that affect you as a caregiver? Is we know that we have to make sure to keep an eye for kids, especially when they're asleep. They're at risk of kind of dropping their oxygen levels a little bit. So that's called nocturnal hypoventilation. And usually that's why we say, how to kind of as a surveillance thing every few years doing a sleep study to make sure that when our kiddos are asleep, their oxygen level is not dropping. Because then there's, CPAP, non-invasive respiratory support that we could use. And these weakness is the cause of one of the major challenges we see in the disease, which are the respiratory complications, like we talk about either with seizures, but also commonly with any kind of respiratory infection like flu or RSV or any kind of viral respiratory infection that can lead to bigger complications. And also for a lot of our kids, they have feeding difficulties, so coordinating and swallowing becomes risky. They have a risk of things going down the trachea instead of the esophagus, and that's called aspiration. And a lot of our warriors need support with feeding, either by modifying the texture of what we feed or supporting with G-tube. Lastly, I think I have imaging findings as a reminder that they can be pretty normal MRI findings when kiddos are young and kind of like newborn, less than 6 month old period. Over time, we tend to see a lot of white matter abnormalities. And so that's what I put this word here, because some of you may have said or may have heard the term leukodystrophy, or there's some white matter component to the imaging findings. A lot of our kids have a thin corpus callosum, and the volume loss is also seen kind of variably, which is what this picture is meant to show you here. If I can put my pointer, that basically the space around the brain is more space, less tissue, which is what atrophy means. I think one question that comes often is, can I look at the MRI and tell you what problems are going to change or are different, right? Like if we don't see any change in our patient, but the MRI is changing, what do we do with that information? And I think that's tricky because there's not a very direct correlation of if there's a change of this much, this is going to affect our kiddos this much. So I think it's a balance of, to me, paying attention to how our warrior is doing and the symptoms, and worrying less about the MRI changes, because sometimes it's not a clear-cut correlation. So for providers and doctors, Emily already shared this, but that's a QR code for the gene reviews. So I think you guys already have that, so I'm going to skip that. So I'm going to transition from kind of like whirlwind of overview of, what does TBCK cause clinically, to next, what are we here for today from the research side, and updates about what many people call clinical trial readiness, right? So how do you kind of pave the way forward in terms of what do we need to do now to be ready for future therapies? I think there is a specific, I try to kind of focus it on what is relevant to us, which are neurodevelopmental disorders, right? So one of the issues to consider that, different than like an adult problem, right? Or like kids of any age, right? Or kids regardless of the genetic background are going to look different at different ages, and they're going to be measures that are developmentally appropriate for a 6-month-old that are very different than a 6-year-old, that are very different than a 16-year-old, right? So we need to think about kids as a moving target, right, in general, and how do we characterize anything that we need to characterize needs to be tailored to that movement. I think the other challenge in the rare disease and neurodevelopmental realm specifically is that there are thousands of single gene disorders. So characterizing every one of them becomes, challenging. I think from the basic science research side, with many of these disorders, which is similar to TBCK, we've only known about them for a number of years. We are still trying to understand what the mechanism is in terms of why does missing this protein or how a genetic variant is actually causing disease. And that's critical to develop targeted therapies, right? Or the other word that you may hear is disease-modifying therapies, right? Which is different than symptomatic therapies. Like we can treat seizures, we can support kids with therapies, but they're not targeted to that specific disease, right? So the targeted therapies is where we want to go. I think we know that there's differences, and meeting different warriors of different parts of the world and with different variants, that there's differences in genotype phenotype correlation, and we know that this system of the same way a blood pressure medication, for example, is tested, is not feasible for rare disease, because we're never going to have thousands of people and doing a large placebo control trial. Sorry, friends. All right, next slide. Do you want to see it, Mama? Okay. All right, so natural history studies. What is natural history and why does it matter? Let me start with that. So natural history is basically tracking disease progression over time just to understand what typically happens. I'm going to have to pause because we have a very unexpected speaker here. Hold on, where do I mute here? Sorry about that. I can't see you guys, so hopefully you're still, your screen share is paused. Can you guys see my screen? I don't know what I did.
We can totally see your screen.
Okay, sorry. We lost a watch and it's a major tragedy. So data collection for natural history, the idea is to try to collect all the symptoms that are associated with the disease, variability, and progression among different patients. And I think the idea is that we can then decide what is worth measuring and how do we best design clinical trials. And that's what the endpoint or clinical outcome measures that you'll hear probably a lot today means. And also that's where like biomarkers come in, like how do we identify something we could track and we can measure. So we'll talk a little bit more about that. And I think, again, why is this particularly critical in rare diseases? Because, again, we're not going to have 10,000 patients in placebo control and 10,000 in medication, and then we can see what works or not, right? So characterizing very deeply the small number of patients we have is super helpful to move forward. Okay. You'll also hear kind of different words to describe the type of natural history that studies. So there's called prospective natural history means, which is the gold standard, basically means starting from scratch, moving forward, and you bring patients in, let's say, to clinic, you're gonna have everybody do the same assessment, you're gonna measure everything very carefully, let's say every 3 months, every 6 months, you're gonna bring patients to do a very detailed characterization. Those are obviously
Soma,
most, yeah.
sorry to interrupt. Can you stop your share and restart it? I think that it's paused and we're not moving forward.
Oh yeah, see, that's what
We're
I said,
moving
something
forward.
weird, sorry. Okay, it says your screen share is paused. I don't know how I paused it. Okay, resume, hold on. I found a button.
There we go.
There we go. I'm so sorry,
I
guys.
think that's right. I'm hoping others are seeing maybe a thumbs up from someone else. Okay, looks great.
Yeah,
Eric gave a thumbs
okay.
up. There we go.
Okay, sorry about that. So
Thank you.
prospective versus retrospective, and I am losing track of time. I should better go like speed it up. Basically means do we start from scratch and move forward? That's prospective versus retrospective, which means let's use what we already have in terms of can we leverage clinical notes and records and all the things that are happening for routine clinical care, right? Like you're already going to the doctor, you're already getting MRIs, you're already getting therapy, but how do we collect that in a way that is uniform and synchronous that it could be examined for everybody's benefit? And I know we are going to have some more about that later on with the citizen platform, but that's kind of just so you know why are we thinking about all those different components. Okay, let's see if we are moving forward or not moving forward. Okay, so next, biomarkers. So biomarkers are measurable indicators of response to treatment. That's the ideal goal of a biomarker. You want it to be something that hopefully correlates with, let's say, disease severity, and it can change over time in the way that the disease is changing. I think it would be the dream if we can get something that you could measure in response to treatment efficacy, right? So patient one receives drug A and you measure something before drug A and you measure after drug A and you can see a change in response to the intervention. Things that are examples of that, for example, will be things you can measure in plasma, in urine, so blood, urine. And I think that's what I highlight, accessible samples, because things, for example, like CSF, like doing a spinal tap every 3 months. Nobody wants that. You don't want that. We don't want that. Doing an MRI every 3 months when it takes sedation or anesthesia, right? Also very challenging. So how do we find something that is accessible? And again, this is important, especially for FDA approval. And when we get to the regulatory time point, because we need to have something kind of quantifiable and objective to measure efficacy of any intervention. So for TBCK symptom specifics, that's kind of like the definitions. I think one important consideration is what developmental scales are most appropriate, right? Because let's say in the neuromuscular weakness world, like muscular dystrophy and all, if we take a playbook, right, like what is already being done, many of those scales we would have what we call floor or ceiling effects, because our warriors may not be able to perform, for example, a 6-minute walk test. That's something that is used in muscular dystrophy. But if most kids are not able to do that, then you don't have any way to measure the range, right? I think things that are probably useful are seizures and respiratory function measures. Dr. Albino is going to be talking later about how do we very intentionally do community engagement. So we also define your voice, like what's important for the community, like what you guys are, are your priorities, which I think is very important too, and accessible data collection platforms like we talked about. I think from the basic science, I'm going to turn off my video because I think there's a very distracting person here. I think from the basic science side, there's a very major question, which is, do we have a therapeutic window? Like, do we need to introduce a therapy within there's a window of time that is best to introduce a therapy, and what is that? And I think that is something that we definitely need to keep improving our animal models so we can try that, kind of figure that out. And also there's different strategies and therapy modalities, like which is the most safe and effective, and towards what symptoms, like what are the symptoms that those interventions could target. Okay. Update on biomarkers, the last part. So I think in the imaging biomarkers for TBCK, personally, I think we are not there yet. I don't know if MRI imaging biomarkers will be feasible, mostly because what we talked about, for most of our warriors, they will need sedation, anesthesia. It will be very costly on a research level to get like an MRI every 6 months on, even 20 kids, right? Urine biomarkers, this is from a paper we already published a couple of years ago, where we have some trends for urine oligosaccharides, and we have been collecting those over time at CHOP. I think it's promising, but it's challenging at the same time because we don't see the exact same signature, even in the same kid. If we follow them over time, they're pretty variable, right? So for a biomarker, you want something that is very consistent, that if I check it 10 times, 9 out of the 10 times, I can see the same thing. And with the urine, it's still a work in progress, but I'm going to share some exciting new stuff we're doing with blood-based biomarkers. So we're doing targeted mass spec for specific glycosylation metabolites, and Dr. Albino is going to talk about a collaboration we have with untargeted. So basically, do I know what I'm looking for, or try to find something new? To try to maximize what we could get from blood. And from the blood metabolites, we already have about 14 patient samples that we collected in the last, so this is new since last time because we just figured out this signature in the last 6 months. We're looking at patients' blood and the fibroblasts that many of you have so graciously donated over time. So this is where maybe something you did 3 years ago and let us do a biopsy is now helping us down the line. And this is one of the very preliminary data I got this week, but we're starting to see that for some of them there's small changes. Control is black, TBCK, everybody together is red, and this is specifically the 126 variant. But basically we can either see small but significant changes, or we can actually see in some of them it's very promising because we're seeing a 30-fold change, right? Which it would be ideal. So we're working on this. The goal will be that by next, hopefully, submitting an abstract and starting sharing some of this with the community, the scientific community, so we can learn more about how to move this forward, and then keep collecting. So we hopefully, I'm hoping we have at least 20-25 samples so we can publish it. And then what else are we doing in my lab? We keep working with the iPSC neurons, which you guys know it's the main project or model that we use. The TBCK story with mRNA trafficking, that's out, available for you to review. This is our QR code if you want to see the manuscript in a bioarchive. It's under revision in cell reports for the science people who know what that means. And then we keep working on the lysosomal and mitochondrial effects of TBCK. We are working with an exciting collaborator who's doing long read RNA-seq to try to figure out how all the different warriors have different variants, and how does this affect the RNA of TBCK, and does that give us different proteins? So that's super interesting, because that will inform what therapeutic modalities are helpful for each variant. And because of these profiles I was showing you, that all points that TBCK must be doing something with the process of protein glycosylation. Could that give us new therapeutic ideas? So we're excited about that. So that's kind of a early but exciting project in the lab. And I just want to give a shout out to the people who are collaborating with us in that. So Miao He, who is a glycosylation expert, and Lang Ling, who is an RNA queen for sequencing and RNA therapeutics. And I think I'm over time, so I'm going to stop there. That's everybody in the lab. And I put in blue the people who are actively working in TBCK, and I'll stop sharing. And then Emily, you can tell me if we're doing questions later or...
We are planning on doing questions for everybody sort of at the end. So if it's all right, we'll go ahead and move on to our next speaker. And that is Igor Ramos. So if you want to go ahead and share your screen. I'm hoping that you're still here. There's so many individuals to sort through. It's hard to it's hard to find you. So go ahead and make sure you
Hi,
come off mute. Welcome, welcome, welcome. We're so excited for you to share your work.
thank you. Hi, everyone. Hope you can hear me okay. Can you see my screen?
Um, Not anymore.
Not anymore. Okay, sorry. Let me try again. Here, share. No, it's paused, just like Zooma's. Just wait a second. Okay. Can you see now?
Yes.
Okay, thank you. So, hi everyone, I'm Igor. I'm a PhD candidate in Maria Rita's lab here in São Paulo, Brazil. And we're really looking forward to share with you what we've been up to this past couple of years. So I'm going to start by introducing our center. We're located at the Human Genome and Stem Cell Research Center here in the University of São Paulo. We are the largest center for the care of individuals with genetic disorders in South America, and the researchers from our center have been providing care to families since the 1960s. And we have a multidisciplinary ambulatorium where patients can go through genetic counseling, molecular diagnosis, be evaluated by different healthcare specialties, and also engage in research and scientific education. And we have been studying all these years neurodevelopment, craniofacial malformations, neuromuscular and skeletal disorders, and also hearing impairment. And in the center, we've come across this family. This is the first family we've encountered with TBCK syndrome, So these are two sisters, and they are compound heterozygous for these two variants located in the exon 23. And a couple of years ago, we've come across these two patients that are homozygous for these variants. This one has a exon 7 to 22 deletion, and this one has a frame shift variant. We were able to establish iPSC cell lines from these patients, but the results I'm about to show you were generated in CRISPR iPS lines. A couple of years ago, we've done our first CRISPR round, and we generated two cell lines. And we've come across these preliminary results that indicate increased proliferation in our neuroprogenitor cells, and here indicated by EDU incorporation. And also, we suspect of altered differentiation because of this increased DCX marker. It's a immature neuron marker, so we weren't able to reach statistical significance, but we see a consistent trend. So we look forward to investigating this further. In the scientific literature, we come across with inconsistent findings and also limited. TBCK function is now well established. We are working. Everyone in this room is working really hard to understand it better, but it's not really established. And also, the neurodevelopmental consequences of TBCK deficiency are also poorly characterized. And as Dr. Ortiz Gonzalez was saying, we don't fully know the molecular mechanism. And if we don't know that, we cannot properly develop therapeutic approaches. So the goal of my current research project is to step back and evaluate the effect of TBCK deficiency on fundamental processes for neurodifferentiation, such as proliferation and the proportion of cell types. So to study that, we've performed another round of CRISPR gene editing. So we chose the exon 23 because it's the location of the variants from our first patients. And we've performed this workflow to target TBCK with the CRISPR machinery. And we've also generated we've generated 3 knockout cell lines and 3 isogenic controls that have gone through all these steps alongside our knockout. From here on out, when you see this symbol, are the letters VZ is the control, and when you see the letter T, the cell line is TBCK knockout. So we've performed this CRISPR validation and characterization by Sanger sequencing. We can check if the mutation we generated is there, and if there are none of target mutations. And we've also done the genomic characterization by whole exome sequencing according to these guidelines from the ISSCR. And all lines were considered good for us to move on working. And as you can see here, all cell lines exhibit the expression of undifferentiated state markers such as SSEA4 and OCT4. And they all, here in pink, they all exhibit decreased TBCK mRNA expression in levels that are comparable with the patients that we already had in culture. So we moved on. And here are our 3 isogenic pairs and the mutations that we were able to generate. And so starting from these iPSCs, we developed cortical organoids that are like 3D structures that mimic the neurodevelopment. It's kind of a mini brain for so I can simplify it. And then we started to evaluate these organoids we've performed the dissociation, and we've plated them onto MEA plates that are, it's a way for us to confirm that they are electrophysiologically active. So these neurons are firing, they're working, and they're functional, so we can keep studying them. And all cell lines exhibit expression of these neurodevelopmental markers, both for progenitor and neurons. So the organoids are developing as they should and as we expect them to. We also evaluated TBCK protein expression in these organoids, and as expected, the knockout they display a really low TBCK expression. And something that stood out for us is that TBCK organoids are significantly larger than controls, and that aligns with the previous result I showed you of increased proliferation. I haven't been able to directly analyze proliferation in these organoids yet, but we intend to do it in the next months. And something I'm really excited about is this single nuclei RNA-seq. It's a really cutting edge technology, and I'm the first researcher here in our center to do this kind of experiment and analyze this data. So this data is really, really preliminary, and we are really looking forward for your input. And if you have any advice for us to how to improve this analysis, it's really welcome. So these are the clusters that I've been able to get. I'm still working on the cell type annotation and all downstream analysis. But what I'm going to show you are these really, really preliminary results of differential expression and enrichment analysis. So these all remain to be validated, and they can change, so don't get really attached. But we think it's important to let you know what we've been up to and hear from you your feedback. So these are the top 10 up and down regulated genes. We haven't been able to dissect each of these genes yet, but if any of these stand out for you, please let us know. We can pay some special attention to them. And here is the preliminary enrichment analysis. Some of these terms are really make sense, right, with what we know of the gene and of the syndrome, such as translation and protein transport, intracellular localization of structures, and also nervous system development. So we're really excited to further explore this data. And these CAG pathways also really do make sense and align with the current literature, such as ribosome and other neurodegenerative disorders, such as Parkinson's, Alzheimer's, and general pathways of neurodegeneration. And our next steps include the validation of these preliminary results, and we look forward to go forward with this analysis too. I'm currently working on this cell type annotation, and we aim to perform this differential expression and enrichment analysis within these cell types. I think we can answer some really cool questions with this part. And we also intend to reconstruct the differentiation of these organoids with pseudotime analysis, and try to understand better how TBCK deficiency impacts neurodevelopment, so we can pave the way to therapeutic approaches. We also intend to do a new batch of organoids to perform validation experiments. And if you have any advice or suggestion for validation experiments, we are really looking forward to hear from you. And that's it. Thank you for your time. And we are really glad to be here sharing with you what we've been up to. I'm gonna interrupt my screen share. And that's it. Thank you.
Thank you. I hope that you can see all of the clapping and emojis coming through. That is amazing work. Thank you so much. remember, everybody, if you have questions, you can throw them in the chat, and our researchers can respond there. We'll also have time in a little bit to do questions, sort of unmuted, and have a little bit of a discussion. Um, Next is Dr. Wilfredo. Um, so if you want to take over, uh, thank you so much. We're really excited to hear about your work in Puerto Rico.
Perfect. Can you see the screen?
Yes.
Okay, nice. Thank you so much for this opportunity. I'm very excited to be here today. As many of you may not be familiar with me, I'm Dr. Wilfredo Jesus. I'm a pediatric pulmonologist here in the island in Puerto Rico, and we work with rare diseases, including TBCK. So this year we wrote a pilot project for funding, and we were funded. So I'm very happy for that, and the work of, I mean, the findings that we may find here in the island with the TBCK community. The title of our presentation is Advancing Respiratory Monitoring in TBCK Syndrome through Non-Invasive Multimodal Assessment. Um, as a summary of need, most of you know that TBCK is a rare neurodevelopmental disorder that impair the motor function, the cognition, and also the respiratory health. So respiratory insufficiency in TBCK is often multifactorial. It means that involves the muscular weakness, the impaired airway mechanics, but also the disruption of the central respiratory control. So when I was looking into the literature about what problems in terms of respiratory issues my patient with TBCK here in the island may may have, I found that like 84.4% may have respiratory complications, that those may include or not ventilation dependency, tracheostomy, the use of BiPAP, sleep apnea. So I was a little bit concerned about all these respiratory issues. So that's why I decided to study a little bit more about respiratory problems in TBCK. One of the issues that I have in my clinic is that I cannot do any pulmonary function in my patients because for pulmonary function, we need at least some cooperation from the patient and to follow some basic instructions in order to blow and to exhale and inhale the air. So we have some limitations in that type. So when we have a patient in our clinic, I could look of the patient, I can auscultate the patient, but I don't have any tests in that I can do to assess what is inside the lung and how the mechanics of the lungs works. So with that in mind, I said, well, let me see if I can find something else that could give us an idea of how we could evaluate the pulmonary mechanics of my TBCK patients. So that's why we started working and addressing these gaps. So we proposed a multimodal effort independent respiratory assessment strategy that integrates three complementary tools. The first one is the sleep study. When we do a sleep study, what we do is to put the patient overnight with a monitor on it. And what we do is that we evaluate everything in terms of how the patient is breathing at night, how the coordination between the brain and the lungs happen. And that's good to know because we could assess if the patient needs oxygen, if the patient needs any respiratory support, CPAP, BiPAP, mechanical ventilation. So that is something that I'm very interested in what will be our results. Also, we have some equipments in our clinic called capnography. It's like a nasal cannula, like the one that you see here, and that what that measure is the carbon dioxide. And the carbon dioxide, we know that if it accumulated in the body, it could harm some part of our body. Also, it could also increase the seizure thresholds. And also, with this machine, we can measure it without the need of a needle. So that's good for our patient too, because sometimes we need to poke our kiddos a lot. And for blood samples. And the real game changer here is this new machine. This new machine is used for asthma patients, for patients with lung disorders. And the good things about this machine called impulse oscillometry is that this machine doesn't really need you to do a lot of effort. It's only a machine that you have. The only thing that you have to do is just put a mouthpiece in your mouth and just breathe normal, like the same way you're breathing right now. So that gives us a better opportunity to explore if our patient with um, difficulties doing the spirometry could also do the impulses oscillometry, and that could help us a lot in terms of how to manage this disease. So what we put together is that this test will characterize respiratory function in three specific physiology domains: airway mechanics, gas exchange, and neural control of respiration. So as I mentioned, this is the machine that is called impulse oscillometry. That's something that you could do all seated in your car seat, I mean, we can use it in different ways. So we could adapt the machine to the patient. Okay. And it measures two specific things: the respiratory resistance, and that means the airway. If there is any mucus plugs or a lot of secretions in the airway, this part of the machine will give us a bad number in resistance. So I will say elevated number in resistance. And that could happen in situations like if the patient has asthma as well, or bronchoconstriction, if the patient have a lot of mucus production, if the patient have airway inflammation for some reason, if the patient have airway narrowing, that also impairs that number. So airway resistance, it could be measured by impulse oscillometry. That's something that we're planning to do. The second thing is respiratory reactance. That means if there is any problem in the lung tissue of my TBCK patient, I can see it through the reactance number. So it's a measure of the elasticity of the lungs. If the lungs are very stiff, that measure could be increased. If the lungs are floppy, also could be affected. So that happens when you have an obstruction, collapse of the peripheral airways. It's also if you have interstitial lung disease, if you have scarring tissue in your lungs, or even like gas trapping. So this machine probably will give us a lot in terms of the airway mechanics and the parenchymal mechanics of the pulmonary mechanics in my patients with TBCK. So these are how the results look like as an example. This is the resistant part. That looks like is the reflection of the airway problem, and this is how the results look when you do a test for lung tissue disorders. What I'm expecting is that these little bars you can do it like at baseline, but also when you do a bronchodilation. Like if you give a therapy to your patient or to your kid, like albuterol, for example, you can measure pre and post albuterol, so you can also have an idea of how the medication change the the problem in the lung, if there is any problem. And usually these dots here, this dashed line is a predicted, what I'm expecting. I'm expecting that all my patients are in this line. But this is what is called the upper limit of normal. If this bar cross the upper limit of normal, that means a problem. And it's a problem irresistant, or meaning airway. And then what happened before and after a therapy, for example. The same here. The predicted is here in the middle of this graph, but then if you have a problem, that lines will cross like the upper limit of normal. So how we do it? So we have a filter, we have a nose clip, and we have a mouthpiece. A mouthpiece is like the same thing when you go to your dentist, it puts something in your mouth just to keep your mouth open. So that's what we can do in patient that doesn't really follow like a specific command or an instruction. So we put all these pieces together, and then we just put these tiny part in the mouth of the child, and we just put a disc nose clip in the nose, and we'll let them breathe normal for 1 minute, and then the test is done. So what um it, we have tested that for, I mean, as part of the pilot project. We're trying to see how feasible is this test for the patient to do it, how they feel during the test, the utility of this integrated approach, and to help us to give a better decision in terms of diagnosis, monitoring, and management of respiratory complications. And so far, we're looking for 12 patients genetically confirmed with TBCK syndrome. We're planning to evaluate these patients in terms of these modalities. And so far, we have a collaboration with the TBCK Foundation in Puerto Rico, so where our clinic is. So hopefully, we can get it the 12. So far, in our clinic, we have been evaluating. Well, we have patients with the TBCK, so we're likely to invite them to participate. So we have some inclusion criteria and exclusion criteria. The inclusion criteria are patients 3 years or older with TBCK that had been stable at baseline. And I mean, we have to wait at least 6 weeks between any respiratory infection. That's an exclusion criteria, and the patient um, have to be without a tracheal cannula. I mean, breathing by nose, by mouth. Okay. So if anyone know patients with these inclusion criteria, so we'll be happy to take a call or write an email and see if we can get the 12 patient. The goal will be to generate the first integrated respiratory profile for TBCK using a non-invasive monitoring protocol. So far, we have 2 patients that have been tested for the impulse oscillometry, and these are the results of a 7-year-old female with TBCK. And the findings, and if you can see these bars here, are before the administration of albuterol, and these bars are also before and after. This is for resistance airway, and this is for reactance lung parenchyma. And so far, um, this patient had a mixed ventilatory defects in terms of problems with airway and also problems with parenchyma. So there was also improvement in the resistance and reactance post-albuterol. That means that probably the lungs of these specific patient have issues with compliance. That is how easy is the lung to expand and get back to the same shape. And also patients with these 2 findings presents in when they have a lot of mucus in the airway. So there is another patient that we evaluated. In this case, was 10-year-old female with also same issues. So both in airway and lung parenchyma. And that improved after bronchodilator. So far we had only 2 patients. I would like to see the data of the 12th patient or more, and to have I mean to get a better approach and better data about these results so far. Um, this is the contact information for our group. The emails I can give it, I mean, to the TBCK Foundation to share with you guys. And we are planning to do a next clinic in December 13th in the morning in Cayey. It's a town here in the middle of the island. If you're interested to participate, just write me or any of the co-investigators, and we're happy to take any question if you want. Thank you.
Thank you so much. I'm sure that you've already shared info on that clinic with TBCK Puerto Rico. We'd also be happy to make some social media posts and remind people about that amazing opportunity to get involved with research. thank you so much for all that you do and for shouting out TBCK Foundation for our part in providing a little bit of pilot funding to help us find some answers for our TBCK warriors. Um, Now we're going to move forward and ask Dr. Albino if she's ready to tell us a little bit about her research that's also happening in Puerto Rico.
Hi, hola. Let me
Hi.
share my screen. My screen.
It's working on it. There we go. Oh, you are on mute, Dr. Albino.
Mm,
There we go.
I am unmuted, right?
Yeah.
Yes. Sorry, I can't see my screen, so I need to share again. Can you see my screen? Now, can you see my screen?
It's working on it.
Yeah, it's loading.
Oh, I
There we
think
go.
I need to turn off the camera because my connection really is poor. So let me see. Sorry again. If I turn off the video and I have better connection.
And we need you to reshare your screen again.
Yeah. Sorry for that, Emily.
Don't worry about it. I feel like they move the buttons around all of the time. So just when you get used to it, then everything's in a different place. That
Okay.
looks
Can you,
lovely. And we can hear you.
Thank you. Okay. So, yeah, the title of my presentation, I just want to tell you today that I am really focused on the community-driven research that I have been doing with the TBCK Foundation in Puerto Rico prior we start our pilot project. So for me, it's really important to find out what the community can say about my research proposal, and I take very seriously their feedback. So I will be sharing today the experience on a community engagement studio. So the title of the presentation is Community Driven Research Priorities. For me, this means we are talking about the research goal that you, the families, have told us are most important. And this is not research about you as a community, it is research with you and for you. So for me, this is very important. Our guiding principle, as I mentioned, we are doing research with and for the community, not about them. So we use a special approach called community-based participatory research, or CBPR. What this means is that we see families, you as expert partners. You are not just people we study, you are the experts on what it is like to live with TBCK. So for me as a researcher, your daily experience is the most valuable knowledge we have. We also know that doing this work in Puerto Rico has unique challenges, and these like funding, finding a specialist, getting access to care, and dealing with complex healthcare system can be very difficult. And our research must be designed to help solve these real life problems too. So our main goal for the first part of this project was to listen. We wanted to systematically identify the priorities, the concerns, and the recommendations from the TBCK community in Puerto Rico. We wanted to know what you think the research should focus on. So this approach is built on 5 key ideas, which you can see on the slide: equitable partnership, community expertise, shared power, mutual benefit, and co-learning. So here's a quick look at our timeline for this work. We have celebrate a virtual call with families. We explained the project, the pilot project that you funded, and we introduced the idea of having a community engagement studio. This was a really good call because we have a lot of families, Puerto Rican families living in mainland, so they have the opportunity to connect too. So in September, we held the Community Engagement Studio. This was focused meeting where we work with TBCK community leaders to co-create the research plan. And next week, I am waiting for confirmation, but next Wednesday, we have a call, another virtual call to start consenting process and give clear instructions for sample collection and interviews. So this is the next big step in moving the pilot research forward. So why study the natural history of TBCK encephalopathy? And I know Dr. Ortiz talked about that in her presentation, but before we get to the findings, let's quickly remind ourselves why this research is so important. So why do we need to study the natural history of TBCK encephalopathy? That was one of the aims of my pilot too. So understand the journey. So we also want to improve or at least demonstrate access to care, also prepare for treatments, build a before treatment map needed to measure if the future therapies work, empower families by offering clearly expectations and reducing uncertainty. We can help families feel more in control and prepare for what may come next, and create hope for the future. So every child's story, every piece of information you share adds valuable knowledge that helps the entire TBCK community find better solutions. An important part of this research is creating a TBCK biorepository. That was one of my aims of the pilot. And this will be like a safe place to store samples, blood, hearing, buccal swabs that can help us learn more about the condition. For the first time, we will be including both individuals with confirmed TBCK diagnosis and their parents. So we will be doing an untargeted approach to see if we see differences in analytes or metabolites from carriers as parents and the patients or confirmed cases. So this will be making this a true family effort in discovery. So why does this matter for me? Because it speeds up discovery. So researchers, not only in Puerto Rico, but globally, can study TBCK's biology more closely to understand why symptoms happen and how they differ between children. It supports therapy development. Scientists need real patient samples to test new ideas. It enables future studies. So the samples we collect today can answer research questions that we haven't even thought of yet years from now. It reduces the burden on families if samples are already available. You won't have to repeat tests every time a new study starts.
And also it builds collaboration. So we can share these samples without any names attached. So with trusted scientists around the world to speed up progress for everyone. I just want to say that our main goal is to honor every family's contribution by transforming your generosity into discoveries that brings us closer to better care and treatments for TBCK. The findings I am about to share come from an structured 2-hour meeting we call Engagement Studio. We work with 3 female parent leaders from Puerto Rico TBCK community. All of them are caregivers of individuals with a confirmed TBCK diagnosis. It was held on September 5th and lasted 2 hours. Importantly, the entire discussion was held in Spanish to make sure everyone was comfortable and could express themselves fully. We used 7 key questions to cover everything from trust and logistics to support and how to keep the project going. So these couple of slides show the 7 questions we ask. They were designed to explore every part of community-centered research. For example, we ask about what makes family feel safe and confident to participate. In terms of logistics, what do we need to consider for things like transportation, scheduling, and access? For data collection, what are the challenges and opportunities for collecting data like surveys or lab work? In terms of support, what extra support like emotional, educational, or financial help will benefit families during the research? For boys, how can we make sure the families' voices are actively included in all the project's decisions? In terms of this dissemination, how should we share the results with different groups like other families, doctors, academic centers? And the last one, in terms of continuity, how can we make sure this project continues, not just in Puerto Rico, but globally for a long time? So the feedback on the study itself was really positive. All the participants rated the engagement studio as a totally agree across all metrics. As you can see here, this tells us that the way we work together, the community-based participatory research model is highly accepted and valued by the TBCK community. Now let's look at the main findings. The community gave us a very thoughtful and complete vision for the research. It falls into 3 main areas. So building trust through reciprocity and respect. The families need transparency about how samples and data will be used. This is a non-negotiable, and this was really straight forward from them. We need to educate healthcare providers to foster empathy and understanding, and we must always recognize families as their experts on their children's conditions and experiences. In terms of logistical accessibility and flexibility, research must be flexible. This means offering things like home visits or using local labs for sample collection. We should offer virtual interview options, and we need individualized scheduling because we know that evenings or nights are often the best time for busy parents. Holistic support beyond the protocol. The research should offer more than just the study itself. This includes things like facilitating psychological service or workshops. The data should be used as evidence for legislation to help with things like medication or transportation access. And we should share self-care tools for parents and caregivers too. So the community also highlighted the importance of long-term vision. So, in terms of authentic community partnership, this means continuous communication and feedback throughout the entire research project, keeping participants informed as a sign of respect. Strategic and multi-channel dissemination. We need to share these results in many ways. This includes creating educational videos for health professionals, to teach them about TBCK and how to work with families. And we also need to promote the foundation's visibility. In terms of sustainable long-term and systematic change, the research should lead to real lasting change. This means these things like providing genetic counseling that is not existing in Puerto Rico and working with integrate research into standard clinical care and policy. So these findings give us a clear, actionable roadmap for ethical and impactful TBCK research in Puerto Rico, and we will integrate flexibility into all our plans, offering home visits and virtual options, pair research with immediate benefits, such as providing resource directory or psychological support alongside the study, formalize our long-term partnership with the TBCK Foundation as equal collaborators, and design dissemination strategies that make sure the results reach everyone who needs them through many different channels. To summarize the key takeaways, the TBCK community in Puerto Rico has given us sophisticated and complete vision for patient-centered research. Trust, reciprocity, and authentic partnership are absolutely foundation of this work, and the research must be sensitive to your life reality, flexible in its logistics, and always aimed at creating immediate and long-term positive change for the families. Finally, this work is a call to action for everyone involved. To researchers, we must embrace these principles and honor the expertise from the members of the community as co-creators of knowledge. To funders, we ask you to support this community engaged research that provides mutual benefit. For example, to policymakers, we urge to see this research data to advance health equity through new laws and systemic change. And to the TBCK community, please continue to lead and shape the research agenda. So by placing your voices at the center, we can ensure that the quest for knowledge is in rare diseases like TBCK, that is very frequent in Puerto Rico, is not only successful, but also deeply unfair and just. So thank you, and I'm happy to take any questions you may have.
Thank you so much, Dr. Albino. The chat is lighting up with people so impressed with how thoughtful you've been with your research strategy in Puerto Rico and how important it is to our community. I do have that video from the Bosch Lab, but I think that we'll just save a little bit of time for that and try to roll right into questions since the chat has been so active. So at this time, if you do have questions for Dr. Albino or anyone else, go ahead and come off mute and ask them. One that was asked in the chat is are the research opportunities that are available in Puerto Rico right now, is there is there a plan to expand them to have them be for non-Boricua variant patients in the future?
Is that a question for me?
It could be for you or Dr. Wilfredo, but I think that he had to step out. So if you want to go ahead and address that, that'd be lovely.
No, I think this will be inclusive. I know that we have a founder effect and we have we as a country have the most cases reported or confirmed. That doesn't mean we need to exclude other TBCK families, especially families from here that migrate to the mainland and for receive services. So I didn't mention it, but in the virtual call that we have on July, we try to plan a strategy to include all the people from mainland too. I know this is a pilot, but I think thinking about the future, it will be really important not just to consider people from US because we are a territory, but also from other countries that are underrepresented in genomic research, such as Latin America. So it will be really lovely to include, for example, um, the patients from Brazil that Igor are studying and other, cases confirmed.
Great. As we say in our community, no one fights alone, and every one of our warriors is important. It's been really great to have sort of our researchers spread out all over the world. So we're so lucky to have such a wide research network. It looks like Dr. Ortiz Gonzalez has a question or raised
Well, no,
hand.
I was just gonna follow up on that. That the hope is that because these are smaller sort of pilot studies, that we can then sort of publish and show feasibility, and then hopefully that can also help us get more funding, because obviously it's much easier when you have this big of an island to get everybody to drive and go get samples and do the respirometry or whatever in like a 3 month period, get all the recruitment you need versus if you're trying to recruit across the world. So we're kind of taking advantage of that as part of the idea. And then hopefully we can get funding for kind of bigger, broader projects. Because I think especially some of these, collaborations, we are thinking about how do we make this kind of start with a small group and then expand it to everybody.
And follow up on that, I think this is really important because we need preliminary data. I should say that at least the results from this community engagement studio will be published, and this really important because this help other researchers to include community in their research and consider them collaborators too. But as Dr. Ortiz said, with this time situation with funding, we need to start small and then uh, start publishing and get more funding.
Um, for something that's known really well sort of in our research community that might not have made it out to our wider TBCK community, there is currently kind of a crisis in funding research in general. A lot of the funding that comes from the US government has been removed. A lot of the funding that comes from different parts of the government, whether it's NIH or the National Science Foundation, has also been removed. And so that means that as researchers for TBCK, it's really important for us to be able to say, hey, look, our research works in this little group. Can you give us some more money so that we can see it work in the wider community in a bigger group? And just like if you were going to a bank and asking for a loan, you're showing them, look, I've paid my mortgage on time all of the years. This is sort of how scientists prove that they are trustworthy to be given research funding. So the TBCK Foundation funds pilot work like what Dr. Albino and Dr. Wilfredo are doing in Puerto Rico. And that's a way for us to sort of take that step and build that research and get additional funding sort of down the line. In the chat from Inez is asking Dr. Albino, will the calls and sort of everything that you've been doing with this community engagement sort of be available, be published, be out there for our community, but also for our researchers?
Of course, yeah. I think the calls will be open and also we can share our resources. so they can be implemented in other places. So I think this will be really, really nice. And also, it would be really cool if we can compare experiences because, again, the context in Puerto Rico will be different from mainland, so or other places. So it will be really nice if we get this feedback and start doing comparisons.
Wonderful. Uh, Dr. Ortiz Gonzalez answered a question actually in the chat, but could you maybe answer that out loud as well, please?
Sure. So I think Ines, sorry, was it Ines? Ines, hi Ines, asked about blood gases for monitoring of oxygenation. And the question is really for Wilfredo. I am not a pulmonologist, as you know, but since he left, I would say that because usually that's a pain prick, and especially if it's an arterial blood gas, which is the sort of gold standard, that's like very painful. We don't use that unless you're like sick. Like it's very much a ER PQ kind of situation when you're not oxygenating, but not for like monitoring. I think the idea of piloting this new technology is that we have something that is non-invasive that you could do in a routine visit, just put the mouthpiece and hang out there for a minute to monitor respiratory issues without having to be invasive. I think Wilfredo also put his email there, so you feel free to email him to see if he agrees. But that's what I would say from like the pediatrician version.
I'll also send him a message and see if he gives us a more specific answer, and we can do a social media post on that. Um, a question that I have for Dr. Ortiz Gonzalez is I've seen in the literature in rare disease that some communities are doing things like nasal swabs, so getting towards this sort of non-invasive. And do you think that that could be something that we could look into as we're investigating biomarkers, or do you think that that's not quite right for TBCK?
I guess I would need to think about what exactly are we doing with the nasal swab sample, because if it's for like DNA, RNA type stuff, I think that's pretty easy and safe. I think for the metabolite type approach, I think it's less helpful. Elena, you have your hand up, so I'll let
Yeah,
you comment too.
I think the answer for me will be it depends what question you want to answer. For example, nasal swabs are using currently for microbiome research more than purely extracting DNA for genomic research. So if I am asked to use nasal versus buccal, I will prefer buccal for DNA for genomics research. But if you want to do nasal microbiome, you should go for the nasal swab. So it depends the question you want to answer.
Thank you. There's a question in the chat about how neuromuscular problems can impact um, TBCK kiddos with hypoxia.
So just to make sure I understand, you mean hypoxia in the setting of like a pneumonia or illness, or like in general kind of hypoventilation, meaning that you have less ventilation, your lungs are less full than usual? For Diana, I think is who asked, just to make sure I understand the question. I don't see her on the.
Yeah, for hypoventilation. Sorry.
Yeah, so yeah, no, that's okay. I just want to make sure that I don't tell you something useless if I don't understand the question. Yeah, so the neuromuscular weakness can affect the hypoventilation, because I think the simplest way to think about it is like if your lung, like what Dr. Wilfredo was talking, right? Like the balloon needs to expand and contract every time you breathe. And the muscles around your chest, especially your diaphragm, is responsible for moving that balloon open and close. So if your muscles are weak, that's going to make that doesn't expand your chest cavity as much. So that's what we tend to see in TBCK, that there's like a, what we medically would say, a chronic respiratory insufficiency, that kids don't ventilate as much at baseline. But then in the setting of like, and that's why any kind of virus or infection or anything that disrupts breathing can make the respiratory complications worse. So the neuro is a bit of a kind of cycle that because of the weakness, you don't breathe as well, but then that also causes other problems. I think the research that Dr. Wilfredo is doing because is to help understand if it's just the muscle that is driving the respiratory issues or is there's actually something different about the lung itself because there's very little information about that. So we are blaming everything on neuromuscular weakness, but we don't know if there's anything different about how elastic, how tight the lungs, like that balloon, if it's a stiff balloon versus a very stretchy balloon, right? That's going to make a difference in how you air moves through it. And that's what we are hoping to learn from the research with Dr. Wilfredo.
Yeah, because her pulmonary doctor said that it wasn't her lungs, that it was mainly her brain not sending the signal.
Yeah, so that's
Mhm.
another aspect. So there's, I think, the neuromuscular weakness is one. Two is your brain telling your nerves to control that breathing rhythm. And three is if there's anything going on with the lung itself. So I think there's sort of three pieces of the puzzle driving the respiratory changes. I think the biggest one is the weakness, but I think we haven't looked carefully at the other ones.
Yeah, because first they told my daughter she has chronic lung disease since she was diagnosed, and then she was on the ventilator, you know, at first 10 hours a day, then 12 hours a day, then 16, then um, now, 24 now. So it seems like as she grew, throughout the years, she just started declining, because she, they get sick with pneumonia, bronchitis, all these illnesses and respiratory problems. And now she just became totally dependent on the vent,
The vent.
24/7. I can disconnect her to transfer. Her saturations stay up, but her respirations are high. That's how that works. And it's like hard to see her, now on this stage, but it seems this is a big concern with TBCK. Like as far
Yeah.
as I have seen, like it's the most concerning part that I have dealt with my daughter. Another thing I wanted to ask, if you don't mind, um, my daughter was put on this seizure medicine called Xcopri.
Mm-hmm.
And so far, my daughter has been without a grandma seizure for 6 months almost, when she used to have 2 to 3 seizures a day, like grandma seizures. Not a
Mm-hmm.
day, I mean a month. I'm sorry. And I really don't know. I mean, I'm seeing an improvement on her seizures, but I really don't know if you think this is a good medicine that could treat kids with TBCK with the situation with the seizures problem. What does it have like in it that is helping my daughter a little bit more?
So
If it's
it's a
something that can
newer,
help.
yeah, no, thank you. Exactly. I was gonna say it depends. The seizure management is a kid by kid situation, and this is why it's really hard to say. I very broadly put it there for a reason because I think like some kids, I have kids that did terrible on the ketogenic diet, and other kids have done great on the diet, right? And the same thing like, it's Xcopri is also known as cenobamate. It's a newer medication. And so that because it's fairly new, not that many kids have been on it. So I personally, I think only know one kiddo that Dina, I don't have to follow HIPAA because Dina already put it on the chat and it didn't work really great for him. So
Yeah.
but if it
It took
works
a
for
while.
her, it's great for
Yeah.
her, right? Like just stick
Yeah.
to if she's telling you that it's working for her, so then it's great. But we can't
Mhm.
like say, oh, now try it for everybody else, right? Because it's a kind of thing that it's we found that it's super variable child by child in terms of the epilepsy. A lot of kids have done great with lamictal. We have one friend in the call that I'm looking at right now that it was terrible for him, and I don't understand why because most of my other kids did great with lamotrigine, right? So it's really unfortunately a case by case scenario, but I'm so happy if it works for her, then that's great, right? Like if then stick to it.
Yeah. I mean, it took a while for it to work. I mean, you have to, increase
Yeah, you have to
dose little by
titrate.
little
Yeah. Yeah.
until you find the right, dose for her. Right now, she's on 300 mg once a day at night, and she has been okay, but it can go up to 400, the max. And
Yeah.
it has been working, but like as parents, we always want to see what is a good medicine that is not gonna, put him to sleep or, relax him
Yeah,
too
and I
much.
think the side effects are also tricky because not everybody, some kids do great in terms of side effects with one medication and others don't. So that's very much a your neurologist one-to-one conversation based on what medications they've been on, which one has worked for them, which ones have not. We would love to get, this is part of one of the things that we would love to get when we get more natural history, like all the history of all the seizure meds kids have been on, to see if we can have more like data-driven answers to that. But so far it seems like it's very kind of individual and it has to be tailored to each
Yeah.
patient. But
Okay.
thank
Well,
you for
thank you
sharing
for
that,
your
because
time.
maybe next time I have somebody, I'll try.
Yeah. Thank you. Okay.
Okay. So many questions coming in on the chat. Uh, Vitalia asked a question of Igor. Um, can we get a little bit more detail about the link to ribosome and your RNA-seq data? Um, any ideas from the individual hits? And I'm asking this one out loud because I had a very similar question about the functional data coming out there. And if you've thought about looking at cell mobility as well as proliferation in response to some of those gene hits that you saw.
Okay, regarding cell mobility, we look forward to investigating it and mostly like neuronal branching and migration. Maybe we're still trying to figure out what would be like the optimal model and experiments to do that, but we intend to do it. And regarding the enrichment terms, these results are really like fresh out of the oven, so we weren't able to explore it deeply. But as soon as we can confirm that they are real and dive a little bit further into them, I'll definitely share with you guys. Uh, I
I mean,
think
it is
that's
very
it.
interesting because I mean the ferry data says that a lot of ribosomal RNA is binding to it, so to the complex. So it's very cool. So nice to hear.
Yeah, that's why we're like really excited to see these terms such as like general translation machinery, but specifically ribosome. So as soon as we finish validating it, I'll definitely get in touch with you guys.
What I'm hearing is that we should do a research roundtable in a couple of months and get all of our brilliant researchers together to talk about some of these big, cool new data that we have
Yeah,
coming.
I'll definitely be there.
Awesome. Thank you. Um, all right, I am just checking the chat. I think that there is a longer question in here for Dr. Uh, Ortiz Gonzalez. Asking, it seems a little bit about gene therapy. So maybe if you're willing, Dr. Ortiz Gonzalez, to uh, just give a quick answer to this question about AAV and gene therapy, and then we'll move into the video from the Bosch lab.
Yeah, which I don't know if that's going to be, there's possibly some overlap with what that's going to be, but I think I put something in the chat. Happy to talk about it. This is like a whole separate conversation. There's a lot to think about that, and we've definitely thought about it. I think the video from the Bosch lab will have some pilot data on their animal model and AAV rescue, which we're all happy to see. But yeah, we're happy to. I think gene therapy is something we're definitely considering. I think in the kind of big picture answer is that TBCK seems to be a dose-sensitive gene, meaning that too little is not great and too much is not great. So when we put it in neurons, iPSC, patient-derived neurons, so far is toxic, which is a problem because when we do AAV gene therapy in vivo, it's hard to target which cells you're going. And I think we need a little bit more understanding of target development and what is the sweet spot for expression. But I will just leave it at that. And then maybe we can see there's something that the Bosch lab has been working on. So I think that also would be super helpful to see.
Awesome. Thank you, Dr. Ortiz Gonzalez. If you have more questions or you're thinking about this later today and have something that you want to ask, you can always send your science related questions or really any questions at all to me at emily@tbckfoundation.org. It's literally my job to track down our researchers, track down the answers for y'all, make the connections that you need. So please never hesitate to reach out there. If Facebook or social media is more your style, pop things in there. Amber and Inez and Nikki and Francis and Lauren and so many members of our community are constantly monitoring in there to make sure that we're finding the answers that y'all need as quickly as we possibly can. With that, I'm going to go ahead and share my screen and hope that this works as flawlessly as I've seen it happen before. Um, Let me know if it doesn't. But this is a video. Unfortunately, the Bosch Lab all had other things going on today and were unable to attend. But when I asked them if they could record a talk, they were so excited about it. So here is Dr. Bosch giving an update from her lab, which has been a recipient of our pilot grant program for the last 2 years.
Hello, TBCK community. This is Elizabeth Boge, and I know I probably met many of you who are attending before already, but for those that I haven't met at previous meetings, I'm an investigator at the Children's Hospital of Philadelphia. I'm an assistant professor at the University of Pennsylvania. I see patients with known or suspected genetic disorders, including those with TBCK syndrome. And our laboratory has been working on understanding more about TBCK for over a decade now. So I am sorry that I can't be with you in real time today. Whenever you're listening to this, I will be in a campsite with 12, 13-year-olds Girl Scout camping outside of Gettysburg. So I wish that I could be with you, but unfortunately I have the prior commitment and can't make it today. But I still wanted to update you with some of the work that's been going on in the lab. Our lab was very grateful to be awarded a $5,000 grant from the TBCK Foundation a few months ago to Rajesh Andretti, a scientist in the lab. And that grant was meant to cover learning more about how TBCK affects specific cells in the brain by using a mouse model that we have used genetic engineering to remove a functional copy of the TBCK gene. And so, like TBCK patients, those mice have a lack of TBCK in the brain, and so they have similar symptoms to what the patients have. So they have seizures, and they have trouble feeding, and they have muscular issues and neurologic issues. So because it's much easier to be able to study mouse brains than human brains, we wrote a grant to use a more advanced type of microscope to look at the mouse brains. And this type of microscopy is called FibSEM. And what it does is it looks at a subcellular level. So it's not just looking at the cells that make up the brain, it's looking at what's inside the cells that make up the brain. And what we're doing is we're trying to find specific differences in the organelles that are in the cells. So just like our bodies have organs, cells have organelles. And so we were using FibSem as an advanced type of microscope in order to learn more about what's specifically going on inside these cells. That is going quite well. The samples have been collected already. They've been processed. And Rajesh gave me an update that this week was the first time that those samples actually went into the FibSem machine for analysis. And so the project has gone all the way from starting with the live mouse to getting them into the microscope. So we're going to have to spend some more time collecting additional data, but the project is well underway, and we really appreciate the TBCK's Foundation's funding of that. One of the reasons that it's important is we still don't quite understand what goes wrong in the neurons of children and adults when you don't have an appropriate amount of TBCK protein. And so if we can understand at a more detailed level exactly what's wrong in the cells, then that can give us better ideas about how to fix it. We're hoping that if we can figure out exactly which of the organelles in the cells look most affected, especially if we look at different mice over time. So if we look at mice right when they're born versus when they're older, we might be able to get more of an understanding about what are the first problems that the cells have, and then what other problems accumulate as the mice get older and the neurons get more affected. So more generally in the lab, I'm really pleased to be able to give some additional updates. We were able actually for the first time at this meeting to publicly talk about some of the targeted therapy work that we've been doing. And so we have two different targeted therapies that we've been working on in the lab. The first is gene therapy. So we've been able to take a harmless virus and put the TBCK gene into the harmless virus, and then put that gene into the brains of mice who don't have TBCK. And we've shown that it's both effective. So if we put it, we can see that the TBCK protein gets made. So if you usually go gene to RNA to protein. So what we did was we put in the DNA, we put in the gene, and then we could see that the protein was getting made. We saw that the mice tolerated the injections of the gene therapy, and it looks like there's a beneficial effect on the lifespan of the mice. So this is the very first step of getting a therapy into patients, which is of course our major goal. And those are the first 2 points that we have to meet, is make sure that what we're doing is, first of all, is safe, and second of all, it's effective. So we're really excited to be able to report that is progressing nicely. The second targeted therapy that we've been doing is looking at changing how the DNA gets turned into RNA, which would then eventually go on to make a protein. So everyone's TBCK protein, it's actually kind of like pieces to it. And there's different ways to make the TBCK protein with using different pieces of the gene. So it's kind of like a mix and match system. We talk about it very simply, like there's one gene and it makes one RNA, and then that makes one protein, but it's actually much more complicated. So our bodies can take one gene and play mix and match with different parts of the gene to make different RNAs that are all different types of TBCK. If you make different types of TBCK RNA, then you can go on and make different types of TBCK protein. And so it turns out that there's a naturally occurring type of TBCK that our bodies make that doesn't include the common 126X Caribbean founder mutation. So the reason that that's important to know that it's possible to make some TBCK protein that doesn't have this mutation in it, because that part the body just skips it when it's using mix and match of the different pieces. That's just a piece that it doesn't use. So what we did was we used tiny pieces of RNA called ASOs, and you can take the ASOs and you can put them into patient cells that have the common 126R mutation, and then it tells the body to skip the part of the DNA that has that mutation. So it's saying, you know what, we know that this mutation is harmful and it'll stop the whole rest of the protein from getting made. And so we can make a different, shorter protein that doesn't have the mutation in it. So it's not changing the patient's DNA. It's not gene therapy. We're not giving more of the DNA or the protein back to the patient. We're telling the body to mix and match the DNA that's already there in a way that doesn't use the parts of the gene that have the mutation. So this is only, this particularly one that we're making is really only applicable for any patient that has the 126X mutation. Even if you have just one copy of it. So I know there's a lot of patients that have 2 copies of 126X. This should be in cells, this is effective for those patients in those patient cells. It would also be effective in patients that only have 1 copy of the 126X mutation. So we are at an earlier stage for that. We haven't tried, we've only tried this in cells. We haven't tried this in mice because our mice don't have that mutation. But we are really excited about this as another type of therapy. And also that you can put that also as a proof of concept, so that if it's possible to skip other people's mutations when the mix and match process is happening, that this might be more widely applicable to other people as well. So we are really excited about the progress. I just want to warn people that just because we have a therapy that works in cells in the lab or that works in mice in the lab, it's still years away from having it be something in patients. And so we're still working towards getting therapy into patients. It is always the utmost concern on our minds all the time. But it's just the first step in the process, and we're all working very hard to bring these things to you as patients and families as quickly as possible. But it is just the first step. But we're excited about the potential for it going forward. Thank you very much. I appreciate your time and your listening, and I was happy to get any questions. And please feel free to reach out if there's any questions that anyone has about the work that we're doing in the lab. Thank you so much.
All right. There was a lot packed into a couple of minutes there. So as Dr. Bosch indicated, if you have questions about what's happening in her lab or any of that research, go ahead and send them my way or to her directly. There is a question in the chat that asks for an explanation of what gene therapy means. Any of our researchers want to try to tackle that? Don't all speak at once. Uh, so right now, gene therapy is sort of a broad term for the idea that when there are variants in our genes, like what our TBCK warriors experience, there's sort of a change in the DNA, which then means that there's a change in RNA and then in proteins that can cause different types of issues. And so gene therapy targets the actual genes, so trying to either take out the part that's not working quite right or add in a part that does work properly. And so gene therapy for something like TBCK syndrome um, makes sense from a science perspective when we're thinking about it. The size of the TBCK gene is amenable to being manipulated, but that doesn't always mean that it works out like that. Um, it's sort of one of those things like Florida is a great place to grow citrus fruit, but that doesn't mean that every year we grow a lot of citrus fruit there because there's a lot of other factors at play. So, TBCK syndrome is amenable potentially to gene therapy, but we need to do a little bit more research, as Dr. Bosch was saying, to really get it targeted in there and understand one, what TBCK, the gene and protein are doing, and then two, whether or not the changes that are possible to be made with gene therapy will actually help in cases of TBCK syndrome. She talked a little bit there also about the timeline. So, if you've seen in the news about baby KJ, who recently at CHOP received gene therapy. What the news articles didn't really talk about, and I did a Facebook Live about this a little bit as well. To give baby KJ gene therapy, so many things, so many stars had to align. He was in the right place at the right time, but also that therapy that he was given that seems to have been very successful was over 10 years in development. So getting the team together in the right place at the right time to create the conditions to help baby KJ. There's a decade worth of research behind that. What's important for our community about that is that baby KJ went first. That was an approved N of 1 investigation. And that means that we watched how that process happened. And so the time between when our research is ready and when our community is ready, we can control that little bit of time right there. And so that's what we're doing by making sure that our community is very engaged, making sure that we're supporting our scientists, and making sure that we're understanding how the regulation of using these types of treatments is being done. So gene therapy may be coming. Uh, Obviously, we're working on it. And hopefully that answered the question satisfactorily. Um, I think that we have all the questions that were in the chat. And this is actually a great segue. So we've done our science updates. Feel free to send more questions as they come up. And now we're gonna just like change gears slightly um, and talk a little bit about how our community can be more involved with research. So, you heard from Dr. Albino about how important it is for her to really understand what the community wants and needs in order to move her research forward. And that's true not just for Dr. Albino, but for our entire research community. So, one of the things that we are initiating is, sorry, I am looking for my share so that I can share my screen on the next part of our adventure here. So, we've already been through our science updates. Hopefully, this is gonna go. Maybe. There we go. All right, we've already been through our science updates. Next, we're going to talk about our research opportunities. I'm just going to switch up our order right here a little bit and welcome Gio, who works with Citizen. If y'all have been paying attention to our social media recently, we've been kind of announcing a little bit this partnership with Citizen Health. And what they are helping us do is a number of things. One, they provide a platform by which we can collect all of the retrospective data. So that's sort of the data that's in your health records, information like Dr. Ortiz Gonzalez said earlier about what types of medications you've used, things like that. That's all in your health records. And our researchers can, uh, if we share that data, can collect that and better understand what medications work best in our community, um, and potentially even what medications might work best depending on variant and things like that. And then there's some other really cool things that are offered to our community because you know that we don't make partnerships unless we're really sure that it's going to be really great and provide a lot of value for our community. So I'm actually going to stop my share and send it over to Geo to talk to us a little bit about citizen health and what besides gathering this electronic health data that platform offers.
Thanks, Emily. I first warning, I am like calling in from my hotel at a conference, so the Wi-Fi might be a little sketchy. Let me know if I'm getting any weird lag or anything. But let me just share my screen here. All right. Y'all can see these slides okay?
Yes, they look
Awesome.
lovely.
Okay. Great. So yeah, it's super nice to meet everybody. My name is Gio. I work on the partner management team at Citizen Health. And I'm just excited to tell you a little bit about what we're doing and kind of the new things, especially. But just kind of quick background information. I think many people at Citizen have different experiences with rare diseases. So for me, I'm a genetic counselor by training. So I've spent a lot of time seeing patients in clinic. But for many of the people at Citizen, that relationship is a lot more personal. So this is a picture of our founder, Nasha Fitter, and her daughter, Amara, has FOXG1 syndrome. So some of these personal connections have really allowed us to understand that rare diseases are really complicated and they pose unique challenges. And overall, we know that these rare disease communities just deserve better and more personalized support, which is kind of what we're all about at Citizen. We also know that for rare diseases, everybody's one of the goals I think that everybody is working towards is drug development, is new treatments. And traditionally, that is actually a pretty hard thing to do. It's not impossible, but it's really hard. And it's hard because there is oftentimes a high burden on patients to collect the data, whether that's attending, different clinics or going in. We also know that drug development is really expensive, and oftentimes, even though it's expensive, those trials can fail. And then lastly, there's really no end-to-end solution. There's nobody that kind of supports basic science all the way through drug discovery and post-therapy tracking. So what we're trying to do at Citizen is really to empower patients, advocates, researchers, drug developers with real-world data insights. And that is all coming from patient-consented data. This is the quick version of the process, but really what we do is patients sign up on our platform and we go out and collect all of their medical records, every institution, every doctor you've ever seen. And we take those medical records and using some natural language processing and some human interventions, we transform those unstructured medical records into research-ready data sets. And we're using those data sets to help drive research. With right now, we'll talk about this in a little bit, but we have had over 25 manuscripts and abstracts that have been published in the last 3 years, just based on citizen data. So while we're doing this whole kind of data transformation process, we're also trying to find unique ways to provide specific value to patients along that journey. There are a couple of core principles that really set us up, I believe, for success in this process. And the most important one is that we believe that patients should have full control over their data. So that means that if you want to just use the Citizen platform to collect your data and keep your medical records in one place and not share it with anyone, that is totally fine. But if you agree to, you can share that information with other people. And that information is all securely shared. It is de-identified. And again, only if consented would we ever share any of that data. And then lastly, because a lot of our relationships with patients and with advocacy groups, we see more as partnerships, like we're working together on this. And so if there are situations where we are able to license that data out to a pharmaceutical company, we also will directly share that data back with patients, or excuse me, share that money, that actual compensation back with patients and with advocacy groups. So I heard you guys talking a little bit about this too, but natural history studies are really one of the first pieces that we're trying to build. We want to understand, how diseases develop and progress. And in some ways, we want to try to understand what the baseline is, like what is an average, or what is normal in a sense for a TBCK. So we're really trying to set those things up so that we can understand with different treatments how things get better, how things get worse, really longitudinally what these different rare diseases look like. And we've noticed that it's this lack of knowledge of natural history that limits our ability to understand the condition. And when we don't understand a condition well, it makes it really difficult to figure out what exactly we need to help treat for patients. Diving in just a little deeper on how this works. So really the goal is that we want to give patients the opportunity to unlock their health data, because many rare disease patients are seeing lots of different providers across lots of different institutions, and even just keeping track of all of that is a heavy lift. So we want to really give patients control over that process. And that's why when we think about all of kind of the different partners in this ecosystem, we really emphasize that you as patients are in the middle. And while we can go out and collect all of your genetic data and your medical records and any imaging, scanning, all of those types of things, and share that back with you, we still think it's most important that you are deciding how that information is shared moving forward. And that could be with pharmaceutical companies or researchers. It could be sharing information directly back with the advocacy groups. It can be sharing information with the new doctors or current doctors or clinicians. All of those things are possible. But again, it really is this patient-driven mission that we're on. Today we have relationships with, now it's actually over 100 different rare disease communities. So really, the vast majority of our growth has kind of been from advocacy group to advocacy group, kind of that word of mouth type of organic growth over time. But we really at this point have had lots of different relationships with lots of different disease areas. I'm actually going to pause this here and keep moving. So really quickly, what this process looks like, and I know that at least a few of you have completed this already, but the onboarding process for Citizen is purposely as painless as possible. So most people get through onboarding in less than 5 minutes, but it really is as simple as creating a username and password, entering in some basic demographic information, and then you are given two very important questions about consent. The first one is, do you want to share your information with your advocacy group? So that just allows us to share information back with advocacy as kind of a hub of understanding some of these rare diseases. But again, that is something that is it's completely optional. Like you can opt in, you can opt out, it's your decision. And then the second piece would be, do you want your de-identified data shared toward research? And again, even if you were to say, I just want to keep the data all for myself, like that is an okay decision. We want people to be able to make whatever decision they want. After you've completed the two consent questions, you enter in the providers. So I saw Dr. XYZ at Hospital XYZ, and through that, we're able to go and collect all of your medical records from all of those different places. And then last, there is a step to validate your identity. Especially for children as a caregiver, that's an important step as well. So this is just a quick picture of what the dashboard looks like. And again, for a patient benefit, this record collection process is free. It costs you nothing. And really, I think, in my experience as a genetic counselor, I have so many memories of rare disease parents coming in with just these binders of information from their health history. And this is like one way to just put all of that in one place. Within the dashboard here, you are able to share the information. So if you were going to go see a new provider, you could share kind of updated information with them. If you had maybe a genetic test report that you wanted to share with a family member or something, like all of that sharing is kind of customizable on the dashboard. And then also we do have the ability to have different, surveys and different ways to touch base with patients. So there is some kind of activity that can happen here as well if you want to participate. And then lastly, once we have uploaded a patient's medical records to their dashboard, we have this really cool new product that we're calling the Personal AI Advocate, um, that really allows you to interact with your medical records in real time. And I want to say that this is new and like almost in beta mode. And when we look at some of these examples, these are really examples that we thought of. So, again, if you're going to see a new doctor, you could say like, can you please summarize all of my medical records into one paragraph so that you can share that information with them? Um, or you could say, I don't remember the last time I had an EEG. Like, am I due for another EEG? Like that type of information would be easily accessible with the AI advocate. But what we're also seeing from patients and caregivers who are already using this is is incredible. Like the ways that people are starting to use it is way past what we had envisioned, which is really, really cool. So this first comment we saw, someone said the AI advocate has been an absolute life changer. I used it to craft an appeal for an adaptive bicycle. Insurance denied it 3 times, and my next step was a court appeal, and then was finally able to get it approved with the AI advocate. So this parent literally was trying to fill out this appeal letter for insurance, went into the AI advocate and said, can you help me write this letter based on these things with these data points from my medical record? And it was able to get that through really successfully. And then the second example, was just a parent who was using it to try to just make sense of thousands and thousands of pages of medical records. And I think that's especially important when some of the unexpected things happen, when you're on vacation and all of a sudden you have to go to the ER for an unexpected problem. Like a lot of people don't know about rare diseases. So to be able to have a quick synopsis or basic information about what's happening has been really cool with the AI advocate. Whenever we talk about AI and medical data in general, like we really want to get ahead of this in saying that security is a significant priority for us. We know that this is sensitive information. We know that we have to be, perfect stewards of that data. And so this is just some of our information about following HIPAA's privacy rules for expert determination, meaning that there is minimum risk. We also are compliant with IRB and GDPR. We have processes and controls in place for Title I CFR Part 11. I pretend to know what many of these things are, though truly don't really understand all of the details of this. But what I would also say is that Devin McGraw, who is our Chief Regulatory Officer, 100% does. So she was the Deputy Director for Health Information for the Office for Civil Rights, and actually was one of the people who wrote many of the HIPAA privacy laws. So if there is a kind of regulatory officer rock star in the world, she would get my vote. Like she's a person who really, really knows this stuff well and allows us to be like very compliant, really up to date when things change. And has been a really, really valuable researcher and partner for us. Really quickly, like we had talked about this a little bit, but when we're able to partner with advocacy groups and partner with patients, we again are able to share back some of that value with them. And to date, we have shared back over $1 million with patients. So I think a lot of times people are wondering, is this just like a couple dollars? But no, in many cases, it's a much more significant amount, just to really appreciate that this is a shared process, this is a partnership, and that there are efforts for everybody involved, including patients and caregivers. Emily had mentioned this too about how important supporting international patients is for the TBCK community. And we do have our onboarding translated to a number of different languages. However, the actual record collection process is frankly much more difficult for international patients because a lot of our processes are built around the laws in the United States. However, for international patients, we certainly allow you to upload your own records. So if you have those and they can easily be added to your citizen profile, that is very easy to do. And that's especially for genetic test reports. I think that's an area where we see, even just one document makes a big difference of us being able to understand these cohorts. So there are definitely ways that we can support the international community as well. And once there are records in the system, all of the benefits of, participating in surveys or registries or even using the AI advocate are certainly available. And lastly, we'll talk just kind of briefly about research. So again, in the last 3 years, we've really been working hard to make sure that our data is used by different researchers. So we've had 11 manuscripts, 25 abstracts, and partnerships with 15 different biopharmaceutical companies. I am going to highlight one here, which I think has been one of our most successful stories. And I'm not sure, maybe some of you have heard this story, but Praxis is a pharmaceutical company. They were running a natural history, study in person in clinic study for SCN2A, and they brought Citizen on really as a partner to help try to increase the number of patients that were enrolling. So that was the goal, was to kind of go wider to find more patients. And what we were able to do was not just find more patients, but we were also able to show that the data that we generate with Citizen is incredibly detailed and deep. And so in the end, once we were able to share this data with them just 6 months after kind of getting things started, they realized that not only were we seeing more patients, but the data that we were sharing with them from our platform was actually more meaningful than the in-person natural history study data that they were working with. So this resulted in kind of the first ever FDA-approved use of real-world data to replace an in-person natural history study for rare diseases. And their estimates were that, for their new drug filing, that this saved them roughly 4 years in that process, which was a really cool, I think, a really cool win for us, and really makes us believe that what we're doing is going to be helpful for other disease communities as well. So that's the end of what I plan to speak about, but happy to take questions if anyone has them. I did include a QR code on the bottom here, so this is kind of the unique link that will get you to the TBCK and Citizen Health landing page if you're interested in signing up. I'll be sure that y'all have my contact information as well. If you have questions, if you want to schedule a call with anyone at Citizen to talk about more details, like, we're very, very, very available. But otherwise, I would love to hear if anybody has specific questions that I could dive into.
Thank you so much. A couple of things that come to mind. It for real only takes 5 minutes to sign up for this, y'all. Like, I've literally sat next to someone while they were signing up in the middle of another conference, and it really did only take 5 minutes, which is amazing for how much information Citizen is able to collect. A couple of extra things for our community. Citizen has let me know on two separate occasions that Puerto Rico is not considered international, which means that all of our Puerto Rico friends can participate completely with this study and not have trouble getting all of their medical records sort of gathered into one place. Um, I know other people in the rare disease community who are using that AI tool to really help with things, whether it's you're in the emergency room and you can't remember the name of that medication that you had an allergic reaction to, and you just like need to check real quick. That AI tool can really save time and effort, and that mom brain of, I'm not sure I remember the 900 million things that I need to communicate in this sort of high stress situation. Let that AI agent sort of help you out there. It can be a game changer, and uh, the TBCK Foundation really wants that tool to help and support our community. And we don't really get anything out of this. So we've entered into this partnership with Citizen Health to collect records and show the community about this because we think that it'll be a really great tool for the community. And before we even said, yeah, this is totally something that we want to do, we asked Nikki Shearer to sign up with Citizen to try it out, make sure it really only took a couple of minutes, and make sure that it was going to provide value for the community. So if you have questions, throw them in the chat or go ahead and come off mute. For our international friends, it's also good to share your genetic report, and that's a great way to do it.
Yeah. So I've got some questions here. Are there any additional complications with adult warriors with guardianship and that sort of thing, or with parents who are divorced, blended families, anything like that? As far as would two parents maybe have their own accounts for like appointments that one was involved in and the other wasn't? Or what are some additional details that might kind of address those types of complications?
Thanks, Michael. So you're asking, just to make sure I hear you, you're asking about kind of two parents having different access to medical or to the accounts, or like providing different information in there. Is that what you're asking?
Oh yeah. And it doesn't have to be different information. It's just it's just that maybe one parent took the child what to one appointment. The other parent took to the other one. Those might not be linked. Maybe they're linked by the patient's name. So that's probably how it works, right? I don't know how that works with granting access. But then also for some of us too, is there additional complications if for adult warriors? So it's different if you're sharing your child's medical information, but is there additional steps if the warrior's over 18?
Yes. So there are different, to the second question, there are different processes for patients who are over the age of 18. And I guess even that is not always straightforward, but I think we have lots of different avenues that you go through, and that's part of the consent, and that's part of the verification at the end of the steps as well. I think we have had other instances as well where kind of multiple family members want to be able to help contribute on the same, child's citizen account. And we have a couple different ways that we work through that as well. There's one person on our team, Mikkel, and she is like, this is literally her job. And she is also a very, very lovely person to work with. But I can guarantee that if that is a concern, like we will either have a solution that works, or because we're like a relatively kind of startup-y type of company, if it doesn't work, like we will find a way to make it work. We're not afraid to kind of to break things to make sure that they work for everybody. So if that's a specific concern that you see when you're signing up, like Mikkel is literally a little chat at the bottom of that. And if you reach out there, she will be able to find time to sit down and talk through all the options. It's a really good question.
If you don't reach out
Yeah,
to
sure.
Mikkel, she'll actually reach out to you. And if you don't respond because it gets like, I don't know, sent to your junk email, then Mikkel will actually reach out to me and just check in and see if I can help make a connection there. I've done this for the other rare disease community that I work for. And just like to provide context, Mikkel reached out to a family who was actually in the middle of a crisis. And Mikkel said, hey, this family is not responding. I'm not really sure what to do. And I responded and I said, oh, that family is actually in the hospital right now. Can we just like press pause on things and we'll reach back out in a couple of weeks and like make sure that they are doing a little bit better before we add something else to their plate. Mikkel said, absolutely, 100%. I will let you take the lead on that. And this was a couple of months ago. We did reach out to the family again after they were home from the hospital. Everybody had slept in their own beds for a couple of days. Everyone was feeling much, much better, and they were able to get the additional forms that they needed. That was actually a case of an adult individual, and they needed that extra, I think it's a guardianship paperwork uploaded into the system. And so it was an easy fix in partnership with people who care and who listen and whose intention is that you have the resources you need to be your own best advocate. Great question. Anybody else got questions? of course, if you sign up with Citizen, you click that box that says, yes, I want my data shared with TBCK Foundation. That's sharing your data with me. Um, and I think I'm the only um, like study director on there. So if anybody else is saying, hey, I want that information, they got to come through me first. And you know how I feel about
Yep,
always
exactly.
making sure that your data stays safe, that your information is used to help our whole community. Because in our community, no one fights alone. And this is a great opportunity to uh, share your information so that we can, get a little bit of pilot data and try to get someone to give us some more money to continue to do more research and understand TBCK syndrome better. Any other questions for Gio before he needs to scamper off to the conference that he's at? Thank you so much for making the time to chat with us today and show us what Citizen can do.
Yeah, it's my pleasure. It actually worked out just perfectly for my schedule, so it couldn't have been any better, but I really appreciate y'all letting me tag along for a little bit.
Wonderful. Thank you.
All right, thanks, Emily.
Um,
Thanks, everyone.
thank you. I will go ahead and I'm going to share some of the data from our community, from our other research efforts and opportunities. So, if you have been in our community for a while, you know that sorry. All right, hopefully I am sharing my screen. And it looks lovely. And this is your Rare X data. So if you were with us last year at the conference in Puerto Rico, you remember me sharing a little bit of data, and that little bit of data was like this much data. And it said that our average TBCK warrior was like I think it was a 10-year-old girl. Um, and we kind of looked around the room and said, hey, that doesn't really represent our community very well. And a lot of that was that we hadn't shared our information. So we have our data collection platform through RareX. You can find it at rarex.org/TBCK. And you can see that since our conference, we've had a lot of people join the platform, and not just our warriors in the US, but also warriors sort of all over the world. Um, and we have a total of 42 different individuals in our RareX data collection platform. The thing is, that the data that we have in there needs to be updated sort of annually. So we suggest that you think about doing it sort of at your warrior's birthday or on TBCK Day, which is in February every year. The reason that we ask people to go back into the RareX platform and sort of redo the surveys is we know that all of our warriors are unique and that they're changing all the time, every day. And in order to capture that information, in order to understand TBCK syndrome, how it changes over time as our warriors grow, or just as things change. Maybe they get on a different medication, like someone said earlier, and the number of seizures that they have drops dramatically. What the RareX platform and what Citizen Health allow us to do is gather that information, take the names and the identifying data out of it so that our researchers can publish on it. And that's really an important step for us to be able to move forward, not just having those little bits of data that prove that we can do this research in order to get more funding to do the research, but also to help us move forward and know what direction we need to move forward into. So if you're already part of our RareX platform, thank you. If you haven't signed up with RareX yet and you have questions or you're thinking about it, go ahead and shoot me an email, ask the questions now, um, or check it out. I'll ask Amber if we can dig up the recording of the introduction of that platform and we can put that out on social media to help people be thinking about participating in that as we move forward. Again, that platform is sort of, I guess, monitored by me. So if other researchers are interested in TBCK community data from that platform or from Citizen Health or from whatever, they need to ask me first. And then I'm saying yes to researchers who plan to use that data to help our community. So you're not just sort of sending your data out there to everyone. We are making sure that our warriors in our community are benefiting from the data that we're sharing. And the data that we share can definitely help our research community do more research and find answers for TBCK. I'm feeling like my screen might be paused. Oh, there we go. Okay. Uh, that is our research. Oh, the one thing that I forgot. So sorry. Um, if you receive our newsletters, you've seen this graphic in every newsletter this year. We're going to continue to have this bulletin board in our newsletters, and we'll put it up on social media. And our newsletters go out via email and sometimes in the mail. And this is sort of just your little check-in of all the different ways you can participate in research. And these don't cost anything, but they do significantly move ahead our community as far as research goes. So we have that RareX platform, rarex.org/TBCK. The Citizen Health platform is citizenhealth/partners/TBCK. And then we also have this study here with Dr. Katsura at CHOP, who works with Dr. Bosch and has been looking at baby teeth. So you can go ahead and contact them if you're interested in sharing teeth. Let's see. Check our time. Check to see. I don't see any other questions in the chat, but does anybody want to come off mute and ask any questions about what we've done so far, about research opportunities, or anything like that?
Oh, at some point, I just wanted to not about research, but I just wanted to mention sometime that we are the TBCK Foundation is partnering with the Utah Kids Foundation, and we've been using that to. So the Utah Kids Foundation has a warehouse full of extra medical supplies, and that was something we highlighted during the gala. We had brought a lot of medical supplies, including G-tube buttons, trait kits, diapers, the Chucks pads, probably anything you can imagine, the gloves, anything you can imagine. We probably got it, and there's lots of ways that you can email me at michael@tbckfoundation.org. Just post something on the Facebook support group, and I'll be putting one today. But every once in a while, I start a thread on the support group and just reply in there, and then I can make sure those supplies I can tell you what we have, and then make sure those supplies get to you.
It's a
Thank you.
really
That's all I have.
Yeah, it's a really cool new thing that we've been doing, and we appreciate that Michael is spearheading this as the chair of our board of directors. And we have Karen who helps make sure that the shipping invoices get paid right away when they come through, to make sure that we can get those supplies that are needed throughout our community in the US, in Puerto Rico. And I think that we've been successful with sending them internationally as well, right, Michael?
Yes, yes. Yeah, we have there were some trait kits. There's some trait kits actually that are on their way to Poland, and there's somebody who's able to get that over the border to Belarus. So even so that's the furthest we've done so far. But hopefully it makes it
Yeah. So
all the
our
way. So
community is strong and connected. Um,
so so don't be afraid to reach out no matter how far away you are. We'll see what we can do.
you can also reach out and get if you do the info@tbckfoundation.org email, that also comes to me, so I can help direct it to whomever it needs to go to from there. So it
Perfect.
doesn't matter who you're emailing at tbckfoundation.org. It's always a person, and we're always going to make sure that you get connected with what you need.
I also wanted to it's Inez, just want to thank Michael because we did recently get in touch with a person in Belarus, like outside of Russia, to get her supplies that we worked on all the way to get shipped to Poland. So thank you, Michael, because that's a long trip. So you're amazing.
Awesome.
Awesome. I guess that's as good of a segue as any to try to move into our foundation reports. So we spent our sort of the first section of our time together today talking about research and research opportunities and getting updates on the science and watching our scientists interact with one another and get excited about the new things happening there. So we're going to switch gears just a little bit here and be excited about what we're doing as a community. So I'll give a short little update on what TBCK Foundation has been doing aside from herding everyone together for this virtual conference. And then we have Francis here to give us an update about what's happening at TBCK Puerto Rico because they are such an important part of our community. And something that I'm not sure gets out to the wider community all of the time is that we have a monthly meeting between TBCK Foundation and TBCK Foundation of Puerto Rico where we talk about what we all need and make sure that we are keeping up our close collaboration that we really sort of kicked off with our conference in Puerto Rico last year. So just so you know that I get to meet with our friends in Puerto Rico once a month, and that's why in our newsletter this year we've had our bilingual page about what's happening in Puerto Rico because they're such an important part of our community. We also have Francis, who serves as our liaison. So our board of directors for TBCK Foundation meets every month and we all get together and decide on things, whether it's that partnership with Citizen Health or it's supporting the Puerto Rico Day parade or it's making sure that we have the funding to do the pilot grant programs and how we initiate that. That group of people did get together once a month, and Frances serves as our liaison between the TBCK Foundation of Puerto Rico and our foundation. So we're really lucky to have her. I'm just gonna move on. So if you've been following along in our newsletters, if you haven't been getting them in the mail, shoot me an email with your address and I'll make sure that you get on the list. But we also send them out via email. And I think that Amber has been creating social media posts with them too, because we've been doing a lot and they're super fun and interesting. We highlight different advocates in our community and things that we've been doing. And this year, what we've added is that we do these quarterly reports. and it really helps ground us and make sure that we really are sort of meeting our mission. So in the first quarter of 2025, we had what felt like 1,000 social media posts, more than one a day for February, which is a short month, but was exciting and um, engaging for our community. We sold a bunch of t-shirts with our No One Fights Alone initiative for this year. I wore that shirt to Capitol Hill to advocate for rare disease patients in general and for TBCK warriors specifically. I had all of your names on the back of my shirt. And like we were saying earlier, um, we need y'all to let us know how we can update our information. So we had 116 warriors identified to be on that t-shirt. And in the literature, there are only 69 TBCK warriors represented. So you can see how we're really like changing the numbers and changing what we know about TBCK because y'all are interacting with us, letting us know that you're there, and we're making sure that the research community knows that you're there, that you're important, that your story and your warrior are unique and interesting and important, and we need everyone to really understand TBCK syndrome. We also love that we get to see that our community is all over the world in 14 countries, in 23 states, plus Puerto Rico. So when we talk about that founder effect in Puerto Rico and that we have a lot of warriors there, that's true. But we have a lot of warriors everywhere else as well. And we are committed to making sure that all of our warriors are represented. As we moved into the second quarter, sort of that summertime, we did the ice bucket challenge and took advantage of Nikki Shearer and I being in the same place at the same time, and we recorded our videos. We again had a ton of social media posts. We announced that as a foundation, we had funded $25,000 worth of research. And you saw the results of that today with Dr. Alfredo and Dr. Albino presenting what they've been doing in Puerto Rico, Dr. Bosch telling us about what they're doing at CHOP. We as a foundation, as a community, as a group, are changing what we know about TBCK syndrome, not just from our patient perspective, but also in research because we're funding that research, we're pushing it forward. And that's not possible without all of you. Our third quarter, it seems, was just as busy. As I was putting this together, I noticed that there was more space for our first quarterly report. And then it got a little bit smaller for the second quarter. And then for the third quarter, it got a little bit smaller still. And what that reflects is not that we're doing less, it reflects that we're doing so much that those newsletters get so crammed full of all of the things that you're doing, whether it's all of the advocacy that happens in Puerto Rico, or it's everyone showing up to the rare disease gala, or it's all of y'all on the move all summer for TBCK and raising funds and doing that, or it's your involvement with our monthly family hangouts. Y'all are doing so much, it's hard to fit it into 4 pages. And so I'm always cramping in this quarterly report, just trying to hit the highlights. Um, so we had over 1,500 social media interactions in the third quarter this year. That means that y'all are engaged, you're doing things, you're talking to one another, you're the experts on TBCK syndrome. And whenever you contact me and you ask a question, half the time I'm referring you to that patient group because y'all know what you're doing. And then I'm on the back end sharing all of that information with our researchers as well. And so we're doing such great work. We're doing it together. And we're really excited for what's coming up in 2026. Um, you're not slowing down, so we're not slowing down. And I am so excited to cram all of the amazing things that we're doing into quarterly reports for next year and to mash all of this together and add in all of the things that dropped off to make our annual report for 2025. It's been a huge year thanks to all of you. And with that, I am going to stop sharing my screen, hopefully, um, so that Francis can tell us more in more detail what is happening in Puerto Rico. You're on mute, Francis. It's that auto mute getting everybody. There you go. I think you're good now. Oh, I can't hear you. And you're not on mute. Uh-oh. I can't hear you. Nope. Mm.
On. 1 second. Okay, so I'm gonna try to make it work while I speak over my phone, which is right next to me, and then I present over my computer.
Okay. It seems to be working just fine.
Okay, now I'm gonna go to the next step, which is sharing my screen. And I am very new at this, so bear with me.
You just watch people who are not very new at this. Still, all of us struggled. So I think that Zoom makes it overly complicated.
Okay,
It's hard on everybody.
I don't feel bad about it then. Okay, just let me know if you can see it, and I'll go ahead and try to move this out of here. Okay.
We can totally see it.
Hi, everybody. So I do have a little bit of an update about the TBCK Puerto Rico Foundation. I didn't prepare one as nice as Emily, just giving you a summary of everything we've been doing this year, but I can add a little bit of information as we go throughout. However, I did not want to lose the opportunity to talk a little bit about ourselves. Like we are a foundation created back in 2020, and it really has the goal of educating, advocating, and providing information to the families that have been diagnosed with the TBCK syndrome in Puerto Rico, which you can see is a large number out of all of the known cases that have been well reported and documented. If you have any information of suspected people that might have the diagnosis, you can always put them in contact with us. We are always welcome to help families, even if they have not been diagnosed. We know the journey into getting a diagnosis is hard as well. Some of them are adults, some of them are just babies, and it can be very overwhelming as we get a good range of people being diagnosed with a TBCK syndrome in Puerto Rico specifically. So you can email us at tbckpr@gmail.com and we'll be there to help you. You can always reach out on Facebook. I didn't put in the Facebook information, but I've been managing the page as well. So you're gonna get someone whether you reach out to us on Facebook or by email.
Now, I do want to also not waste the opportunity to encourage people to be part of our team. We do have a lot of goals, and really having people backing us up, whether you are a member of the board or whether you're voluntary, it really helps us a lot. We have been doing a lot of planning on a lot of activities. So if you're a member, you'll be able to help us in the planning of those activities. You can also help us assign funds, like how are we going to be using all of this money? We kind of get to prepare a lot of the things that we post online, and we get to do very fun donation campaigns. And this year we have a few things cooking as well. And we do want to make sure that if we get more people being part of our team, it really helps us divide and conquer, and we might be able to work on a few things that right now are mere ideas because we cannot just move past that point of, okay, we know what we want to do and we have someone that's going to help us plan it. Keep in mind, a lot of us are full-time, we're in the working field, so we're full-time people, and then we go back and take care of our families and our kiddos, that you know how complicated it can get at times with hospitalizations and just overall taking care of the health and appointments. So anyone that really join us, don't be concerned that, oh, I'm only gonna have like an hour every month. Like it does make a difference into our team that is really based out of parents right now that live through what TBCK is. And for example, me, I live in the US, but I've been hand in hand helping the TBCK Puerto Rico Foundation as well. And I started as a volunteer. I started as a volunteer to plan the event last year, and then I just couldn't leave. So that really just shows how you really decide how much time and effort you want to put into it. And it all starts by being volunteers. But if you already know you want to be a member, we will also open, we're also open for that. When it comes to volunteers, it's really more about, once we plan the different activities or the events, it's going to be very specific tasks that we're going to be needing help with. We also prepare a lot of info tables. That is something that we pride ourselves on. And I'm sad that I don't have like a chart showcasing that, but in every newsletter, we highlight how many info tables we've been a part of, and we share pictures and things like that. It makes a big difference in Puerto Rico just to talk about it, as our presence in social media gets better. So really every like, every share, it makes a difference. But in case we don't get that, we do need to have presence, and our presence has been very focused into info tables, and we're just very lucky that we are able to get into most, if not all of them, for free. So it is really just the volunteer effort of our team that really makes the difference, into some of them taking days off from work just to go and do these info tables. So if you get a chance at any point, just thank our members for taking off their time with their families and work just to do some of this work that has been making a difference. We also work on meetings with families and doing just campaigns. Some of our campaigns are more online, but we also want to move a little bit more into just doing like very official events. So we do appreciate the team also in the US that came up with the idea with Ines and Dina of attending the TBCK Puerto Rican Day Parade in New York. Like a lot of those donations, thanks to TBCK in the US, came directly to Puerto Rico. So us teaming up since last year, from the conference time around November of last year, it has really helped really cement how are we going to move forward as a group of, we're here to not only represent TBCK, but we're also here understanding what is the need in Puerto Rico specifically, what is the need in the US, and how we can help each other meet the goals, because our goals are different, but at the same time, we are very together into presenting ourselves as a unified fort into what do we want to accomplish. And right now we know funds are going to be that one very critical thing that we're both going to be needing and we're both going to be asking. So we try to team up, getting events out there and getting campaigns that are not necessarily going to infringe into one another. And one thing that they have helped us in the TBCK Foundation in the US is selling our t-shirts. Like right now we had one person just dealing with all of the shipping, and right now you can go into the Bonfire webpage and not get like a regular t-shirt. There's different styles, there's different colors. So it has really helped us expand into selling the t-shirts outside of just people in Puerto Rico, bringing them over and driving them over to the person buying it. So it has helped streamline that process, and we are super happy as our design this year was made by one of the parents. He hand draw it, and then we actually put it into a t-shirt and put it into stickers. So we do appreciate everyone for doing that. We do have some other activities that are already planned. In November 10th, we have our limit date for designing Christmas or New Year's cards. You can design your card, and then all of the cards that are submitted by November 10th, we are going to be sharing those with by mail with our TBCK families in Puerto Rico, and we're also going to share those with our sponsors. So that is something we have planned, and we are looking forward for anyone that would like to give themselves the opportunity to sit down and create a fun design for a Christmas or New Year's card. We also have a meeting scheduled very soon with Dr. Albino just to talk about the study and all of the logistics. And of course, everybody that is diagnosed with the TBCK Boricua mutation, we're going to share that information with you as well. The call is going to be recorded. We're still aligning with the date and the time, but please just stay in tune with our page in Facebook, where we communicate all of our news. So if you haven't followed us yet, just make sure you find us. And then back in December 10th, we're having another Zoom meeting. As we get to the end of the year, we know how stressful it can be. So last month we did a mindfulness activity instead of just joining into our regular monthly Zoom call and only just having like a free topic or a topic about epilepsy. We went and did like a mindfulness study for the families that joined. And then for December, we wanted to keep it also in the creative side, where we are going to be doing a creative topic. And it's probably going to be around journaling, and I'm going to be the one presenting it. My first time doing it, so I will appreciate anybody that would love to join us. And we're doing this again. Last year we did Parranda virtual one on TBCK at January of 2026. We know we like to share our culture with everybody around the world. So last year we connected with a lot of people in the island from the US and worldwide, and we wanted to make something fun for the families. I mean, sometimes during the holidays it can be very stressful. From how complicated it is to get toys, to participate into a lot of the family activities or activities you just want to join at. So we do like to make something just for us, where we're just playing music and you're in the comfort of your home and you can join virtually. So we are planning for that again this year, so plan on joining us. Now, a few of the reasons for being a donor or being a sponsor for us is it really helps us identify kids in Puerto Rico specifically with possible diagnosis of TBCK. We can post a little bit more, we can reach out a little bit more, and we might be able to go into different platforms that we haven't tried before. It also helps us get funds assigned to specific families that are just having specific battles when it comes to hospitalizations that are just very hard. Like in Puerto Rico, for example, every day you have to pay for parking. And sometimes we don't think about those things, that it doesn't happen in all of the hospitals in the US. You have to get your food from outside, and there's different rules that you have to follow in each hospital. So that has become a very common topic, that we want to be there supporting our families when they are in very specific needs. We also take the opportunity to do some education in Puerto Rico about the TBCK diagnosis. And at any point, if we have to pay a fee, sometimes it's $20 just for entering, and sometimes it could be more. We haven't experienced that. But in case we have an opportunity like that and we don't have the money or the sponsor, we do want to make sure we don't lose opportunity to continue educating about it, because it has been very important when we go talk to people and they hear, this is the first time I hear about it, or actually I think I might know one or two families that might need a diagnosis on TBCK because of the commonalities that they see. So that really helps a lot. And one thing that people might take for granted is they always ask to see the kids. They want to see the pictures. They want to connect with us. So every time you share a picture of your kid and you share it in the public forum, we appreciate that a lot, because when we go on these info tables, that's what they ask. They want to know these kids, and they want to connect with them and their families. So I do thank everybody that has been sharing their pictures in the public forum and tagging us so that we can repost. And then last but not least, we're always there to help people with quality of life. We do want to make sure that people know what are the different services. And we want to make sure that we can bring the right resources, especially on those monthly meetings. Like parents indicated epilepsy, and we were able to sit down and connect with families, not only in Puerto Rico, but in the US. What other options are there? Let's sit down and only talk about that topic, like ourselves, all the information we have each collected and the experience, which is very good to help other families as well. And we've been able to get resources for like that transition into adulthood. That was the other topic. Families were very concerned because it's a complicated process. So we were able to bring a resource into those meetings to really educate the families and help them become very knowledgeable as to what are the steps that we need to take. Because believe it or not, sometimes it starts as early as 12 years of age. So there are a lot of things that we need to be planning for as families, even when sometimes we can be affected by the potential outcome of our kids. There are adults are out there, so we do want to plan for that adulthood. And there might be a lot more that are just not diagnosed because they just haven't been able to get the tools or the information at hand. And remember, this TBCK well, I forgot the name now, syndrome, is very new. So it's not only ultra rare, but it's also very new out there. Now, if you are interested in donating, we have our PayPal account, TBCK Foundation of Puerto Rico, PR. And then ATH Móvil, which is a very common in Puerto Rico, is TBCK PR. I did wanted to share some of our new donation programs. The one on the left, we haven't started it yet, but we are getting ready to do so. We want to do like a scratch and win type of deal. So it's a little bit fun. Maybe you can carry a card with you into family events and they can just scratch off and have the opportunity to donate whatever number appears on the card. And then there will be a maximum of how much money you can donate. And then in the middle, you would know what is it that you're going to be helping us collect through donations, through the just filling out the whole card. And then we do want to also have like a fun thing where if you scratch the little heart, then you'll probably get a prize. So we are just running those ideas through. So stay tuned to see what we're going to have and how we're going to work on implementing something like that. And also thanks to some of the activities like selling of the t-shirts online, going and meeting, teaming up with the TBCK in the US, and also joining efforts into the team that work with the Puerto Rican Day Parade in New York. We were able to get a lot of funds through that otherwise we wouldn't potentially have. And thanks to those, now we have a program that's going to help us match donations to a maximum of $100 per family per campaign. The campaigns actually last a whole week, and we know when families need help, they are going to be reaching out to us or to their family members directly. So instead of potentially only getting $20 for a family member, you could turn those $20 into $40 by just donating through the TBCK Puerto Rico campaign. So we do want to make sure that people is aware of that, because thanks to the donations that we've been getting this year, we are able to expand our program. Where normally we'll just do a campaign for the family and whatever comes in, we just hand it off to the family. We'll be able to do a little bit more matching. And as we get better, that maximum amount could reach from $100 to a little bit more. Any questions or comments?
In case, I think that Francis did an amazing job. But just so you know, if you buy that TBCK PR t-shirt through our bonfire, the proceeds from that go to TBCK PR. It's easier for us to sort of have the one bonfire site, but then quarterly I go in and see how the sales of that shirt has done, and then I just go ahead and send that money to the TBCK PR PayPal account. Uh, on our agenda for 2026, we'll be asking the board of directors for TBCK Foundation if they want to do that matching donation for the month of June for TBCK PR. So what happened last year is that we did, I think the Puerto Rico parade was like towards the beginning of June. And so what we did is we said donations that were made for the 2 weeks before and the 2 weeks after the parade, we split the amount of donations that were received by TBCK Foundation in half and gave half to TBCK PR. So that was decided on by our board of directors, and I have that on the agenda for the new year to check with the board of directors and see if they agree that that would be a great thing to do again.
Yeah.
TBCK PR does so much for awareness for TBCK syndrome. It's so important, and we try to take every opportunity we have to support them as well.
Yeah, and we did the same thing with the gala. We did a silent auction, and then half of the proceeds and the donations also went to TBCK Puerto Rico. So really, thanks to those two activities, we were able to really initiate this program, because we were like, whoa, that was a good amount of money that came through, and we do want to pass it along to our families when they are in need, and we already run campaigns for them. So let's just go ahead and expand it.
All right.
Very good.
It's time for us to move on, and we're going to, it's my pleasure to introduce Lauren, and Nikki, who are going to tell us about our new Ready for Research fundraising campaign and initiative at TBCK Foundation. So I will stop sharing because I think that Lauren has some slides.
Yeah. Let me pull them up really quick. Okay. Can you guys?
I can totally see your
I
screen.
can see it.
Oh. Okay.
It's Canva.
Sorry. Are we in? It's not doing my.
I think you need the purple button that says.
Oh, there it is down there. We got it.
Yeah.
Okay.
I had the same problem with Canva this morning.
Okay, well, yeah, nice to meet everyone. I'm Lauren O'Donnell. I am new to the board of directors. I'm also a TBCK warrior mom. We are unfortunately in the hospital right now, and just I understand that hospital life, like someone's going to come in right when I'm talking, and that's just the way it goes. So I apologize if there's like a little interruption. But we're here to chat through our Ready for Research campaign and fundraising behind it, and just tie together everything we have just heard from this amazing conference and where we're at with research. And as we all know, that a lot of that research comes with needing funding, and how we as a community can work together to have one goal and further this along. So this next screen, just kind of what this means. We're all working together. We have one goal, and like Emily has said multiple times, like we've all said, we are in this together. No one fights alone. And I just want to re-emphasize how important the data collection is as we move forward with fundraising. The natural history study, all of those platforms that we have to really bring the data piece of it will move forward, like just as important as our fundraising to find gene therapies to move forward with research. It's truly such a vital step, and I just want to reemphasize that all of our warriors are equally important for gathering this data. So doesn't matter the type of variants they have, etc. Everyone's just as important in this data collection. So please consider like adding in your warriors information and moving this along. Our fundraising goal this year, specifically to this campaign, is going to be $125,000. It's going directly to this research. As a community, every single dollar counts. And again, I'll say it a million times, no warrior will be left behind, whether you're raising $1 or $500. It's all going to the same cause. It's all going to moving this data along in hopes to find a gene therapy or research for all of our warriors and families. I wanted to touch on storytelling, and when you're looking into creating fundraisers and even just sharing information, how important it is to make people feel connected to our cause, whether that's through personal stories, pictures, experiences. That's my daughter, Emery. She is 2.5-year-old little warrior. And I think even a simple post or a picture like that brings people together for a similar cause when you see something that someone's going through. And it's also fine to not put your personal stories and experiences in there and just start to educate your community and your family and friends, and just bring connection and awareness to TBCK. That's such a big part of what the TBCK Foundation is about, just bringing awareness. And I think there, like Frances just said, there are so many people that are not even diagnosed, and through storytelling and engaging our community, we can just bring together people, awareness, education, and just bring trust and awareness and credibility to our foundation. So when someone maybe is thinking about donating or has, their company is like, oh, do you have a cause you want to donate to this year? And they've heard of TBCK, they understand what it is, and they've seen a personal connection. It's going to be a lot more enticing for them to want to donate. We have added some fundraising tips. So, explaining why. Did I just stop sharing my screen?
No.
No, we're good. We can still see your tips.
Okay. Inform people why it's important. Where will this money go? I think specifically to what we're talking about with the Ready for Research. It says it in the name. We're using money for research to further along where we're at. And Nikki and I were chatting a few days ago, and just like so incredible. We've had so many campaigns and fundraisers and all of these things, and it might feel daunting at some points to be like, oh, we're asking for money again. I think it's really exciting and important to bring awareness to the fact that this is now like specifically going towards research. All of the money and donations that we've had in the past have led us here. And I think that just gives incentive and brings more awareness and comfortability of like people's donating money is actually going somewhere, and it's actually going to exactly where we say it's going, driving our foundation forward. And just make it as easy as possible to donate. So I have QR codes on the next few slides, direct links. Amber is so incredible on social media. It's so easy to go in and just share those posts. Those go so far. And I don't want to just go over everything Francis was going over too, but even just sharing a post or tagging something can go a long way. We don't, it's hard to even understand how much reach social media has. But if I post it and someone else reposts it, like that's hitting a lot more people than you might even think. And then word of mouth, emails, flyers, ask family and friends and local community members to help share your information. I know personally, we've had so much success just with our friends and local community of bringing awareness. I have had shirts for my family and friends, and my sister-in-law like makes it a point to always wear her TBCK awareness shirt to any of her daughters' doctor's appointments, just so people see the name and they are understanding it. So your family and friends want to help, and it's sometimes hard to ask for donations and to spread the word, but I think it's so important to get that bigger reach. Some fundraising ideas: host a sale. I know Emily chatted about a fun lemonade stand idea she had, hosting a bake sale, or even shirts. Host an event. I think I've heard Francis host a craft night, the bingo card she just showed, a kickball game, a 5K, a dinner event, whatever, your community best, and what they'll engage with. Some things that I have found, I did a fundraiser last December, and I just went to a few local places that we attend fairly regularly and asked if they'd be willing to host something. And I was shocked with how many people were so willing to donate 10% of the food sales that day to TBCK or host us for free for that night. It definitely takes some work, and I understand being a TBCK family, it's not always the easiest to add something else to your plate. So you can go as small as reposting an Instagram post, or you can go as big as hosting an event, and it truly all is so impactful and so huge to our community. Another thing that I think is new and something that I'm starting to look into is a business roundup. I don't exactly know what to call it, but if you've ever been to like a Ace Hardware or a bigger company like that, and when you close out and it says like, would you like to round up and donate to this cause? A lot of businesses are willing to add TBCK as their business roundup. So when people are checking out, they might not have any idea what TBCK is, but they have the option to round up $1 on their sale. And just going through local businesses, bigger businesses, whatever we can to just find little avenues to increase fundraising where we're not always targeting, our specific community, but having ways to increase fundraising that way. And lastly, there are business donation matches. So some larger companies, whether it's somewhere you work for or a family member works for, will match donations typically it's at the end of the year, but it could be any time. So if you donate $200 and you want it to go to TBCK Foundation, some companies will match that. So the TBCK Foundation will actually get a $400 donation. I had a family friend do that last year, and their $1,000 turned into $2,000, and it was such a huge way to increase our impact when it comes to fundraising and making even a small donation go a little bit further. So there are just so many ways and so many avenues to go where you don't always have to be, asking your friends and family, the same people, that might get cumbersome and feel like you're kind of asking a lot. I know sometimes that's how I feel when we're doing fundraising, but there are other avenues, and I just encourage us to work together to be creative and know your community, know the surrounding people, and do something that even just brings awareness. Even if it doesn't raise $1, it brings awareness, and that's almost just as important. And just having us all work for the same goal is going to bring us so much further in furthering TBCK research and gene therapy and just support for our families. I've added the ways to donate here. So the actual GoFundMe link is on the left. QR code, super easy. If you want to take out your phones now, you can easily scan it. This will take you directly to GoFundMe. This is also associated with our 501c3. So if people donate to that, they are able to claim that on their taxes. So I always mention that to people if they are wanting to do a donation like that. And then the next QR code on the right side is directly to the TBCK Foundation. When I am doing something for fundraising, I always try to make it as easy as possible, give people many different avenues to donate. If it's a one-click, one-stop shop and they're able to donate, it's going to go a lot farther than them having to type in TBCK Foundation, try to find the donate button, so making it as easy as we can, finding the links, posting this on your social media or hanging this on a flyer is such an easy way to allow people to do this at their own pace if they want to or not. And just a thank you to Emily for getting the GoFundMe. It was a challenge to get it for TBCK, but we finally did. So we have that extra avenue for donations. So I encourage you to share this, print it. If you need us to email it to you, we're happy to. I have Nikki, myself, Emily, and my dad, Eric, who is also part of the board of directors now. I have added all of our emails. So please feel free to reach out to us with any questions or ideas that you have. And if you need support or you might not know how to fully facilitate it, we're more than happy to be a sounding board, give you our opinions, give you all the resources that we have. And yeah, move the needle forward as best as we can, and together, further research and help our amazing warriors and families. But does anybody have any questions? Or Nikki, Emily, do you want to add anything?
You did a wonderful job. Thank you so much. Lauren is our new chair of fundraising. So, like Francis is our liaison to TBCK PR, Lauren is our chair of fundraising for our board of directors. So she's really put a lot of work into getting this up and running. Um, and I just have a couple more slides. I see that people are wanting those QR codes. Amber is literally blowing up my phone right now, asking for them because she wants to get them onto social media. I promise that they will be on social media later today. You can still always use our normal links on our website for fundraising as well. I know some people aren't super into using GoFundMe. So if you have someone who's like, oh, I don't like GoFundMe, we have other options, as Lauren indicated. We also have a PO box, and you can find that info on our website, on our social media. It's on our emails, on the back of all of the newsletters that y'all get, the postcards that you get, things like that. So if people want to just send us a check, that's also a completely reasonable way to donate, and more some people's style than others. That business match, like Lauren said, it's also sort of a huge thing. You never know what your company might be able to do. Um, I'm just going to go ahead and share my last couple of slides here because we're kind of winding down towards the end of our time together. But this is really has some ideas on how you can help. So we tried to make sure that today we covered sort of things that the TBCK Foundation does for y'all, but we also know that y'all are motivated to be involved. So, how can you help? Well, spreading awareness is huge. Uh, Everybody has seen our sticker campaign. Our infographics are also really big and a really great way to spread awareness. As Lauren said, as Nikki said, sometimes it's as easy as wearing your TBCK t-shirts and things like that and having people ask about it and telling people about it. That's such a huge part of building what we do together. Another way that you can help, like Lauren mentioned, is sharing your data with us, whether it's via RareX or via Citizen. That's We understand that that's a time commitment, and we get it, and we try to make it as easy as possible for y'all. But sharing your data is really like so important to helping us get to the next level with things. We also, you can help if you want to sponsor an initiative. Here are some things that you can do. If you say decided that you wanted to sponsor the sticker program next year, I mean, you might have to fight it out with my kiddo because my family does really love that that's how we support TBCK Foundation. But if you want to sponsor stickers, then we would be happy to set that up. We would create an icon that says that like our sticker campaign was sponsored by you, your business, your family, um, whomever. We also have the translation service that we used here. It's called Wordly. You could sponsor that for the year so that every hangout we do, every meeting that we do can be translated not just into Spanish, which we've been doing in the past, but into whatever language you can think of. Importantly, after this meeting is done, Wordly has been recording everything that's been said, and it creates transcripts that can then be translated after the fact. So I can take the transcript of this meeting from English and put it into Spanish or Russian or whatever, so you can reach out for those, and I'd be happy to share. We also have our newsletters, which we send out digitally and actually in the mail. It costs us a bit to do that, but we think it's really important. We see a lot of engagement from the community, and something that I love is to pull up a Zoom call, whether it's with Nikki or Amber or new families in the community, and in the background on their bulletin board, on their refrigerator, is a postcard from us or a newsletter or something like that. I think it's a good way for us all to be connected. We also have the infographics. Like I said, if you want some, send me an email and I will pop them into the mail to you. But you could also sponsor that for $1,000 a year. That makes sure that it covers the cost of postage and printing. As Lauren mentioned, you can also fundraise in your community with a lemonade stand or with like peer to peer birthday fundraisers. So I'm sure you've seen them on Facebook. You can actually create a fundraiser on Facebook and say that the proceeds go to TBCK Foundation, and then our PayPal giving fund makes sure that that happens. All right. I think that that is everything I have for today, other than saying thank you to you all for being here. We had over 50 people here today, which is amazing, and we're so excited to have shared so much about TBCK with so many people. Uh, Importantly, all of this wouldn't be possible without the amazing board of directors that we have. I saw Dr. Ortiz Gonzalez, I saw Abadias Diaz, um, Cindy Lung, Francis, Lauren,
Eric.
Eric, Michael, so many members of our board of directors were here to support y'all today and to be such big parts of our community. And this stuff's not possible without their support. And then the TBCK Foundation team. So Karen and Nikki have been approving the content of this meeting, have been meeting with me weekly to make sure that we keep everything going. Karen makes sure that we're able to claim a GoFundMe and that all of our paperwork is in order. Nikki is the touch point in our community for everyone. She is always there to talk to our TBCK families, to talk to you, to let me know what else we can do for the community. We have Amber furiously taking screenshots and photos of this event and our other events and posting a million posts on social media, including the last minute ones that I send her at like 10 o'clock at night and say, hey, can you do this tomorrow? We have Inez, who is coordinating the Puerto Rico Day Parade, does our mental health Mondays, and is so active on our social media, especially today. Thank you so much, Inez, for managing everything that's been happening on Facebook while we've been here in this Zoom and participating back and forth. None of this happens without that team, and none of this happens without all of you. So thank you all for being here. If you have any other questions, if they come up 3 weeks from now, whenever, it doesn't matter, email Emily at tbckfoundation.org or any of the other email addresses that you've seen today, and someone will get back to you. Oh, I just got a question about refrigerator magnets. I did just put another order in for the magnets. That is still part of the sticker initiative, so I will reach back out, Erica, when I get them. And if you're ever wondering, I think that Amber will be posting, we do have new stickers. And Amber will be making a post about them, including our new Ready for Research sticker. So if you want to tell your whole community that you are ready for research, like all of our TBCK warriors, send me an email and I will send you the stickers. Thank you all for being here. I hope that you have a great rest of your Saturday. Since this is recorded, if you have any questions after watching the recording, all of those email addresses are still good. Just go ahead and email us. Thank you, everyone, for being here. And we're going to go ahead and sign off. Thanks, everybody.